Yeah, welcome you all to this workshop. We are extremely excited. We have an amazing list of speakers. You can see their names here at the top of the slide. This is just a brief introduction, just to present you the people that have organized the workshop. There are Alex Espinos and Raquel Garcia Hernandez, who will be helping in the chat, monitoring the questions, etc. And then, of course, Ben Hayden and Paul Cisek, who not only most organized the most important parts of the workshop, but also edited this special issue that we will be talking about today. Here I'm just showing you the papers, the titles of the papers that are in this special issue, but just for you to have an idea of the type of topics that are covered in the special issue. So our main goal in this workshop is to discuss as much as possible about these topics. We prepare a program that allows for a lot of time for discussion. So you can see that we have separated the program in two sections. We will have an introduction by Ben Hayden about the special issue. Then we will have Lisa Feldman Barrett, Luiz Pessoa, and Barbara Finlay, followed by 40-minute discussion, then a break, short break, and then Joseph LeDoux, Paul Cisek, and Patricia Churchland, followed by another 40-minute discussion. So lots of time for discussion. Please don't hesitate to ask your questions, to make your comments. We are really looking forward to your views, because we have seen that there are many different fields represented in the audience. So you can see systems neuroscience, computational, Evo, machine learning, but also there are many other people that are working in very interesting fields that I tried to summarize with this cloud of words. Well, you can see that there is psychology, cognitive neuroscience, there is also philosophy, just for you to have an idea of the type of fields that people work on. So how can you participate? It is relatively simple. So you should be seeing something like that. The screen is somewhere here, and in the bottom you have the chat, you have the raise the hand, and then the question and answer button. You can click, type your question, and most importantly, or also very important for us, is that you can vote questions so we can know what you guys are interested in. So we can ask the speakers, or we need to check whether the time allows us to invite you on a stage, and you can make the question directly. So this is all. Actually, I will let Ben go ahead. Just let me mention that if you are interested in this type of topics, you can follow us on Twitter. We would like to thank University of Minnesota for letting us use this Zoom webinar, and then the Instituto de Neuroscience in Alicante and the Barrett community in Barcelona for the help. So as I said, there is time for Ben. I will stop sharing and let Ben talk.
All right. I think can you hear me, and can you see my slides?
Yes.
Yes. Okay, great. Yeah, thank you, everybody. I can see that the room is growing at exponential rate. That's very exciting. The reality is this is the first ever, and as far as we know, the last ever, although, who knows, depending on the demand. This workshop came out of two kind of different things. First of all, it came, as Manuel mentioned, it came out of this special issue that Paul Cisek and I edited over the last several years. It was a lot of fun. It was completely new thing for me to be an editor of a journal, and you had all the headaches of getting people and doing the timing, but you had all the fun too of choosing who was going to write it. And I want to say, both about this workshop today and about the issue, that we did not do an exhaustive list of everybody who's thinking about the brain in terms of a evolutionary perspective. We just did. We just were really scraping the surface. So there's a lot of people, and since this issue came out, we've had a lot of really positive feedback and a lot of really nice things that we've heard from a lot of people who are fellow travelers in this world of looking at the brain from the perspective of evolution. It's been really, really exciting, and I think it bodes really well for the future of the field. Just to give you a very brief introduction in the couple minutes I have before the first talk starts, I will say that the watchword of the special issue that we edited, and probably for this workshop as well, is this expression that was really can be traced back to 1973. Nothing in biology makes sense except in the light of evolution. We really have to understand evolution to understand everything. So in the kind of, Paul and I wrote a kind of introductory article for the issue called Neuroscience Needs Evolution. And the argument we make in the article, we make the argument that fundamentally we tend as neuroscientists to think that the brain is sort of this perfectly designed static thing that was given to us, and there's nothing wrong with it at all, and everything is sort of optimized. And even if evolution is important for developing the brain, none of that really matters, because evolution has also made sure that all the kinks have been worked out, and any kind of residue of the past is erased from what's happening in the brain right now. We think of it like this sort of watch here, but that's just not true. It's obviously not true. There's a whole bunch of kludges. There's a whole bunch of bizarre, weird things that have to do with our history. It's all path dependent. And we make a point that really was made by Paul Cisek in an earlier paper that we got to reiterate, which is that when we think of evolution, we tend to think of natural selection, and we tend to ignore the other part of it, which is descent with modification. And the descent with modification, probably from the perspective of neuroscientists trying to do a better job doing neuroscience, is actually more important. In other words, we really need to understand the past in order to understand the present. The most famous example of this that everybody knows, and we use it also because it's such a compelling example, is that the eye of the human, which evolved separately from the eye of the cephalopod, is really inelegantly designed. And there's a whole bunch of really unfortunate things about it. But as with everything else in evolution, we're constrained by where we were in the past. We're constrained by where we were as a species we were yesterday, and those guys yesterday were constrained by the day before. And ultimately that resulted in this really nasty kludgy thing of a reversed photodetector system kind of going backwards in the eye. And that's sort of the watchword for the way that evolution can help us understand the brain more broadly. I put this here just to talk about the fact that we as a species, are also not sort of evolved to be perfect. We come from a bunch of other primates that have a lot of the similar issues and a lot of similar problems. Ultimately, you put it all together, and we can take the past, we can take our knowledge of the past, we can improve our knowledge of the past and help us answer a whole bunch of questions. I've listed some of the areas here where understanding the past can help us understand the present. But really, rather than listen to me kind of pontificate, let's go straight to the talks. The people who organized this, including me, we all kind of put together our minds, and we decided to take some of the writers of articles from our special issue and some other people who we think have a similar mindset, who may not have been in the issue due to oversight or due to busyness or anything else. And we put them together, and we just want to give them free rein to talk about their views on the brain without giving them any guidance, and hear what they have to say on this topic, systems neuroscience through the lens of evolutionary theory. So let's get started.
I think that's my cue, right?
Well, actually, I wanted to just introduce you first.
Great.
Lisa Feldman Barrett, many of you may know her work on emotion, very seminal work on emotion. She is a distinguished professor of psychology at Northeastern University in Boston, and the director of the Interdisciplinary Affective Science Laboratory. And her work focuses on understanding the nature of emotion in the brain, approaching this question from evolutionary and developmental neuroscience, cultural evolutions, and systems engineering. And so Lisa is one of the people invited who was not part of the issue, but sort of has always been talking about these kinds of things. And so we wanted to get her to give us some perspective from her own work on many of these same issues. So anyway, so thank you for accepting our invitation, Lisa, and please go ahead and share and give your talk.
I will.
And
Thank
her
you.
talk is entitled, oops, I've lost my titles.
That's a fun. I'll put it up for you. That will
There we go. That'll help.
help. Yeah. There we go. You should see the screen now. Is that right?
Yes. Her title is The Role of the Body in the Evolution and Workings of the Vertebrate Brain, or Some Stuff I'm Thinking About. So I'm very, very curious to hear it. Thank you.
Thank you. Thanks so much, Paul, and thanks to Ben and Manuel for inviting me and organizing this. And I'm a little bit the odd person out here, I think, because I've been interested in evolution and development and so on as a guiding principle for a long time, but I haven't actually worked in this area. And I've been very fortunate to have been tutored by Barb Finlay. So many of the ideas you'll see are sympathetic, I think, with hers, and whichever, whatever mistakes that I end up making are really all mine, since I'm really at the beginning of this journey. So when it comes to understanding, brain function, many of us who are speaking in this gathering today have written about the problems of being led by intuition, by subjective experience, or by folk psychology. We all advocate for some kind of evolutionary approach instead, but evolutionary approaches differ. And so I'm just going to list a few conceptual tools that ground the evolutionary approach that I'm developing in collaboration with Barb. First, I'm using a kind of a souped up extended evolutionary synthesis that is souped up with a focus on the animal's ecological niche and the brain's role in regulating the body. I'm trying to place an emphasis on computation rather than content, meaning I'm resisting mosaic approaches and discussions about specific adaptations and behaviors. And when I do consider those, I'm considering them in the broad framework of trying to understand computation. So for example, I have a focus on the brain's predictive architecture, which can be described as various compression versus inferential gradients that compute features. So I don't think of the brain as a feature detector. I think of it as a feature computer. I'm not actually a computer as in a digital computer, but like a computing features, basically forming features. And that the computations in question really create domain general functions in a psychological sense, or to use Michael Anderson, the philosopher Michael Anderson's term, reusable parts. So brain parts provide a certain computation, which is then used to construct multiple behaviors in multiple psychological events. And this framework is an extension of the constructionist approach that I've been building in psychology and neuroscience for the past 30 years, which itself uses a constructivist philosophy of science, or what's been called constructivist epistemology or natural constructivism. And so my presentation today is primarily meant to be a thought-provoking sketch, in part because the framework that I'm developing requires answers to some questions that nobody has asked yet. And the questions that have been asked, the answers are distributed across silos like embryology, physiology, signal processing and engineering, evolutionary and computer and comparative neuroscience, and so on. And so at the end of this, 20 minutes from now, if you're thinking, I have no idea whether she's right, but this is a really interesting way to think about brain function, I've never thought about this before, then I've accomplished my goal. That's really my goal for today. By souped up extended evolutionary synthesis, I mean, I'm thinking that brain functions emerge within a biological architecture that's forged by at least 3 interacting evolutionary mechanisms. That is natural selection on specific genes. So like Hox genes would be the example, or the neural crest in vertebrates. Indirect natural selection that keeps in place organizational or developmental rules like Barb's translating time model, in which general brain scaling functions are related to the duration of neurogenesis. And then there are epigenetic wiring, experience-based wiring aspects. So your brain wires itself continuously to the physical and social world around you, but it also is wiring itself to the physical reality of your own body. And this is one piece that I think is often overlooked in neuroscience, actually, and psychology generally, that a brain first and foremost wires itself to the physiological reality of the body it's in as that body is moving around in an ever-changing and only partly predictable world. I think a key computational challenge of any living creature, and I'm not alone in this, is to coordinate its internal parts and regulate its internal state in a metabolically efficient way as it continues to interface with the world, or at least the parts of the world in time and space for which it has sensory surfaces and that are relevant to its survival. So its ecological niche. And you can think about the evolutionary history of the vertebrate nervous system as about the ways in which the feedback control of this internal state are more and more spatially extended further and further, as well as into the external world, as well as temporally extended further and further into the past and into the future. This is consistent with something that Paul has written. It's also consistent, I think, with Malcolm McIver's very cool work about what happens when vertebrates move from the sea to land, and the corresponding expansion of the visual system, visual range, and the expansion of brain size. So the guiding sort of focus that I'm going to emphasize today is that, or my guiding focus is that a brain's most important job is coordinating and efficiently regulating the many systems of the body. Other scientists have proposed other candidates for the brain's most important job, like consciousness or vision, but I would argue that none of these other jobs is important if you're dead. So I would suggest, like other neuroscientists, that sensory processing is in the service of motor control, but skeletal motor control requires visceral motor control, that is control of the viscera. And endocrine system and so on. And I suspect that this critical coordination is at the core of every important brain computation and the functions that those computations give rise to. So whether you buy this reasoning or not, I'll suggest that placing evolution, the evolution of the vertebrate central nervous system within the context of understanding evolutionary changes in other body systems reveals something important about brain evolution that maybe makes brain function easier to understand. And I just want to say up front, I'm not trying to reduce everything to the regulation of the body. I'm just focusing on this aspect because I think it's been overlooked, largely speaking, in a lot of other work. So for example, consider the evolutionary innovations found in the vertebrate neural crest. Here's a quote from Henry Gee in his book, a recent book that he just published, where he wrote, perhaps the single most important evolutionary novelty in the evolution of vertebrates is the neural crest. So important, in fact, that vertebrates might even be defined by it. And he's saying this because the neural crest, whether operating alone or providing instructions to mesoderm and endoderm tissue, produces not only a brain with novel forebrain, but it also produces novel visceral systems, including cardiovascular system and enteric nervous system, novel sensory systems like the visual system, a lateral line system, an auditory system, and so on, a face and a head, and lots of other goodies that are important to vertebrate functioning. Now, there are genetic differences between somatic and visceral neurons, but the two motor systems, the skeletal motor system and the visceromotor system, are not embryologically and evolutionarily separate. They're actually really entwined. And so we really have to get out of the habit of just referring to the motor system as if that means skeletal motor, because that's not really correct. And these two systems are not functionally separate either. And so here's just one extremely cool example that I like, which comes from Peter Strick's lab work he did with David Leventhal, where what they did was they injected rabies virus into certain visceral organs and then traced its transport across synapses. And they discovered that primary motor cortex contains a visceromotor map. So for example, the trunk representations in M1 and M2 are major sources of sympathetic nervous system control of the adrenal medulla. And a few years later, they found something similar for the sympathetic control of the gut. And such findings, I think, indicate that cortical areas, which are responsible for preparing and generating skeletal motor movements, are also a major source of central command that controls the sympathetic effector organs, suggesting a link between the descending control of skeletal muscles and the regulation of sympathetic output. Interestingly, some of these connections that provide this link are absent in rats. And these experiments that were performed by Peter and his colleagues were actually performed on macaque monkeys. In addition, a primary visceromotor control region, which is the anterior mid-cingulate, is also considered routinely a premotor area in the skeletal motor system. So again, we see that there is this co-localization of visceromotor and skeletal motor commands, at least at the level of the cortex, that might provide at least one mechanism for coordinating the two motor systems. And I'm gonna give you some other examples, some other provocative tidbits from viewing the brain through the lens of body regulation. And that has to do with the hippocampus. And these are literally what I'm showing you here are my working notes. Because like I said, I'm really just at the beginning of this journey. And I'm also not showing you all the references because they would take up too much room. I'm just using illustrative references. But if you're interested in a full reference list, I can, provide you with something longer. So the hippocampus, this is the hippocampus in a box flow diagram, signal flow diagram. The hippocampus is thought to be crucial for episodic memory, for cognitive mapping, and for event coding. But the hippocampus also regulates the autonomic nervous system and the endocrine system through its connections to the hypothalamus via the mammillary bodies. And in addition, it is bathed in reafferent sensory information that results from those visceromotor changes. And that comes from the vagus through the nucleus tractus solitarius to the septal nuclei, which then projects widely within the hippocampal formation. So these viscerosensory afferents are called interoceptive signals. These are ascending interoceptive signals from the vagus. And some of these signals actually regulate eating. Some of these signals from the vagus that indicate the glucose status of cells in the gut actually regulate eating. So this is what I think is the really cool part. The hippocampus, in addition to receiving, well, let me say it this way. The hippocampus, some of these sensory reafferents are about respiration. So the hippocampus is receiving information about respiration from the vagus nerve via the septal nuclei. In addition, it also receives direct input from the olfactory bulb, which it is related to, as you'll hear, evolutionarily and developmentally, it's related to the olfactory bulb, and you'll hear more about that from Barb. These signals independently influence event segmentation and sequencing in the hippocampus. That's the wrong paper. This is what happens. This is just a note to everybody. Don't tweak your talk at 1:30 in the morning. It's a really bad idea. So this is the paper. This is the wrong paper. I'll show you the paper. It'll come up eventually. But the point is really that there is actually really good evidence that these signals are actually helping to create sequences of temporal envelopes that represent the statistical regularities of in the periphery, which means outside the skull. So what they're doing is they're helping to coordinate the hippocampus to integrate internal state information with signals from the external world. And so the core computation here is not really episodic memory, and it's not really cognitive maps. It's something more basic related to event segmentation and sequencing in relation to states of the body. And this envelope probably in humans lasts for about 20 seconds if the patient HM is any, who didn't have a hippocampus is any guide. And I think that this idea that I'm suggesting is really consistent with the very lovely paper by Malcolm and Barb in Paul and Ben's special issue, which I'm showing you here. This is the paper which talks about olfactory and respiratory signals influencing the computations within the hippocampus. Now, I should say lots of brain regions can be described as integrating internal and external state information, for example, like the superior colliculus and the midbrain. But I think what's notable here is that in mammalian brains, at least, much of the time, they use memory to anticipate the needs of the body and attempt to meet those needs before they arise, which is called allostasis. And so memory has a very, very key allostatic or visceral motor regulation function. And memory, when understood in this way, is a generalization from the past to the future based on some features of equivalence, features that are, similarity, features of similarity. And the hippocampus, along with the olfactory system, may support this generalization by creating these features. And this is very consistent with an updated view by Jay McClelland and his colleagues, his complementary learning systems theory. And what I'd like to suggest is that these features that the hippocampus is computing may guide the cerebral cortex in constructing prediction signals with many more sensory and motor particulars from the past, particularly those that provide a nearest neighbor mapping of the body in space, at least in the lateral parts of cortex. And there's a really important maybe just gradient there between lateral and medial portions of the cerebral cortex that we could talk about. But I think the key thing is that at least some of these features are likely to be features of generalization, that is, features of equivalence, are likely to be interoceptive or visceromotor in nature. But you would know them probably in the literature by their psychological names like reward or goals, which is actually the term that Jay uses in his paper. So now let's for a moment consider the cerebral cortex. It's common to use the properties and arrangement of neurons to understand the flow of signals across cortical areas. And doing so reveals a major architectural gradient in the cerebral cortex. This gradient, oh shoot. Okay, like I said, yeah, I need to fix something, sorry. Okay, now we're good. Yeah, this gradient has been verified in many important ways, as you can see here. Maybe as you can't see here. Let me back up so that you can see it. I'm sorry. This is me without having had a full cup of coffee after a very late night. Luckily, this is just a workshop. Okay, here we go. This gradient has been verified in many important ways, as you can see from this image. And it's common to think about this gradient in a number of different ways. For example, you can think about it in terms of lossy compression versus inference, where you're going from cortical areas with smaller neurons and more neurons with fewer connections that are computing the features that are very close to the sensory surfaces, and that across this gradient there, that information is being compressed and reduced in dimensionality until you get to multimodal summaries. Or you can think about it in terms of, and then you can think about inference that going in the other direction. That is going from these summaries to infer the details. You can also think about it in terms of feed forward and feedback signaling, or you could think about it in terms of prediction and prediction error signaling. So what are the regions at the top of this hierarchy? The ones that are receiving the goals and features of equivalence from the hippocampus, and the ones that themselves are computing compressed multimodal features, which is just another name for abstraction or conceptual features, that are in effect the initial features of cortical prediction signals. Well, Carl Friston and his colleagues used the Feldman and Van Essen model of the visual system to answer this question. And Markov and the Kennedy group have their own distance model that they use. But I think neither of these actually includes the prefrontal areas that are most important for regulating the body. They just literally exclude these areas. By contrast, Helen Barbas's structural model does include these key visceral areas. And her lab has accumulated supporting evidence over the past 30 years, working across a number of different vertebrate species, mostly mammalian species, to show that empirically that you can describe the flow of cortical signals as prediction and prediction error by looking at the laminar differentiation in both the origin, the cortical origin of connections, and the termination of connections. Whereas the Feldman and Van Essen model, for example, only considers termination, and for that reason has a lot of error, what are errors, like things that you wouldn't predict, violations of prediction. And what I would say is, I don't know that there's a really good explanation for why this model works, although you could speculate. But this is the general model, that prediction signals or feedback signals flow from cortical areas with less differentiation to cortical areas with more laminar differentiation, and prediction error signals flow in the opposite direction. So why is this important? It's important because it tells us what's on top, at the top of that compression gradient or that predictive hierarchy. And the answer is, it's the cortical areas with the smallest amount of laminar development, laminar differentiation, which happens to be visceromotor control regions. So in plain English, visceromotor regulation is the driver of prediction signals in the cerebral cortex. Skeletal motor control signals, you could think of them as a backseat driver, and sensory processing is really just along for the ride, or maybe it provides a little navigation. What you're looking at here is an intrinsic brain system that contains these visceromotor control cortical regions, these very powerful predicting regions in the cerebral cortex. And I'll just explain what you're looking at. What I've done is I've taken a brain and cracked it open along the midline. So you're looking at the medial views. And the colored blobs here show connectivity, not brain activity. This brain system controls your autonomic nervous system, your immune system, your endocrine system, and so on. And as you can see, it's widely distributed throughout the cerebral cortex. It also includes key brainstem nuclei that are known to be important in visceromotor control, although we often talk about them, as fight and flight circuitry and so on, but it's essentially visceromotor control. It also includes the hippocampus, and it includes the cerebellum, both of which have their own compression gradients and also can be understood in predictive terms. And if you're a neuroscientist who does brain imaging research, then you probably recognize that this system actually contains two well-known intrinsic networks, the default mode network and the salience network, which both go by many, they have more aliases than Sherlock Holmes, these networks, but these are their conventional names. And these two networks overlap in densely connected hub regions that are considered to be the backbone of communication throughout the brain. These are findings from a 3 Tesla magnet. We've also replicated this in a high precision 7 Tesla magnet. And these networks not only regulate the body in every moment of your life, but they also participate in a multitude of psychological phenomena, as you could see here. This is a summary of data that I compiled nearly 10 years ago from 6,000 published papers. Findings like these again reinforce the hypothesis that everything your brain does, psychologically speaking, it's doing in the service of regulating your body. So what does all this mean about brain function? I don't know yet, to be honest, because I'm really at the beginning of this journey. But I think what these findings mean is that what we need is an evolutionary framework that includes visceromotor control and its relationship to sensory processing and to skeletal motor control. And that's what I'm in the process of trying to form an evolutionary framework that includes the interior systems of the body, and one that we can hopefully use to ask better scientific questions about basic brain computations and the brain functions that they give rise to. Thank you. Sorry for the glitches, by the way.
Well, thank you very much, Lisa. I think there's probably some virtual way everyone can applaud by clicking some kind of a icon in the chat. But anyway, thank you for a great talk. I just wanted to add real quick before I open up the question. I wanted to add that in our special issue, we actually have a paper by Lucia Jacobs.
Yeah,
that discusses, exactly that very intriguing point about the hippocampus and
respiration. Yeah. So I think there's a lot more to say about that and about where these findings are come from, and they focus on, you know, theta oscillations, and there are some questions that particularly my engineering colleagues have about, and Barb also has questions too. So therefore I have questions because the people who know more than me have questions, but have about these theta oscillations and their what they mean. But I think it's pretty clear now from a number of different lines of work that the brain is being entrained. Brain signaling is entrained, even sensory signaling, right? Sensory sampling and even the computation of low-level sensory features is entrained in some way to respiration by these two pathways, which contribute independently actually to influence hippocampal firing.
Yeah, there's also the work from Yuri Buzaki's lab, I believe, that also shows the close integration with hippocampal rhythms and autonomic function.
Yeah.
So we don't have a lot of time for questions if we want to stay on schedule, and we do. But again, there's going to be an open discussion at the end, and I think in that open discussion we can really have many of these questions asked. I'm sorry, I'm just looking at the upvoted question. There's a question by Robert Barton, which is a great question, but I'm wondering whether that would be better for the open discussion, because it's a general question. So maybe we can come back to that. And if there's something a bit more, so there's one question from Juan Balcazar that says, is reward defined simply as an external signal arising from internal computations by the hippocampal complex and cortex?
No.
What kind of signal is it? I am interested in hearing the computational definition for the signal that we call reward.
Yeah, so I mean, one of the things that I was thinking of doing when I was thinking about what to talk about in this talk was propose some of the computational systems or learning systems that might exist in the brain. And I guess what I would say is, I think that words like reward and value and motivation trouble me greatly, because they, for example, what is reward prediction error? It's interoceptive prediction error. Now, it might be interoceptive prediction error that is very abstract, that is a multimodal summary, but it's still linked to the state of the body. And I could give you a lot of examples that reward signals are conditioned on the brain's modeling of the state of the body. But you know, you could just imagine the last time you ate too much chocolate. You know, at the beginning, chocolate's wonderful. If you eat too much, you don't want to see another piece of chocolate again. It's no longer rewarding, even though its features have not changed. What's changed is you, that the way that your brain is computing the features that represent that object. But what I will say is that I think that you can find what you would call reward signals in various computational systems in the brain. But there's also, I think, a system that is, you know, souped up, essentially, and that would be, you know, it's sort of souped up and focusing on what we would think of as reward or reinforcement, and that would be the basal ganglia. And so what I'm trying to say here is reward signals are not in any one place in the brain. They are features of many of these computational systems. And the way I think about it is that the cortex, the hippocampus, the cerebellum, the basal ganglia, the midbrain, these are all, you could describe them as integrating over the same kinds of information, but weighing certain features more versus other features. And then they're communicating with each other. So a question like where I want a clear computational definition of a signal that we call reward would be a whole brain signal. It wouldn't be, you know, in one particular region of the brain. And you'd have to consider different computational factors, because what the cerebral cortex is doing computationally is not necessarily the same as what the hippocampus is doing or what the cerebellum is doing. And I'll just say one more thing, which is that at the outset of this for me, you know, because I was sort of looking at this through the lens of predictive processing, I was thinking, well, there's compression gradients and predictive processing in several of these, you know, different brain regions. So does that mean that the brain is running a single model, or it's running multiple models, or, you know, is there one set of signals or multiple sets of signals? And I think what I would say is that there is a brain-wide rewards representation of reward, the features of which are computed differently in these different systems, which have different computational constraints based on their cytoarchitecture and so on.
Great answer. Yeah, so I think in the interest of time, we need to move forward, but it's actually a pretty good segue, what you were just saying, probably, I'm guessing, to some of the things our next speaker is going to say. Our next speaker is Luiz Pessoa, who is a professor of psychology at the University of Maryland, College Park, and director of the Maryland Neuroimaging Center. His research focuses on the interactions between cognitive and emotional brain system. And Luiz is actually an old friend. We both studied together computational neuroscience under the direction of Stephen Grossberg. So we come from sort of philosophically aligned viewpoints. And I think I think I will agree with much of everything he says in this talk. Anyway, so thanks, Luiz, for contributing to the issue and for agreeing to take part. And his talk is going to be titled "Refocusing Neuroscience: Moving Away from Mental Categories and Toward Complex Behaviors." So go ahead, take it away, Luiz.
Thanks, Paul. What screen are you?
Right now, we're looking at your presenter view, so you have to do the little switch thing.
Oh, you are seeing the presenter view, and now you see.
Now we see your slides. That's perfect.
Okay, all right. So now it flipped back to. Okay, let me flip my camera back to where it was. Okay. Since I can't see anything in the audience, let me know if my camera is pointing, I don't know, somewhere completely random.
It looks good.
All right. Okay. All right, so let me start by acknowledging my collaborators, the ones who actually know about the comparative neuroanatomy parts that I'm going to be talking about. Loreta Medina and Esther Desfilis from University of Lleida in Catalonia. I've been working with them for a few years now and learning a tremendous amount. And so this is joint work that we've been doing that I'm going to be discussing here today. So how do we study the brain? If we think about it from the standpoint of, let's say, teaching or reading books about the brain, a very common way to study the brain is essentially to go and subdivide it into a set of domains that have been used for many decades, if not centuries, if not millennia, in some cases: perception, attention, memory and learning, language, motor control, executive functions, and the like. So this exercise that we're engaged in is essentially one, let me just put a timer here so I'm not too off. So this exercise that we're interested in is one in sort of describing these mappings between these domains, let's say perception, and what parts of the brain are engaged in perception, attention, cognition, emotions, and so on. The general claim that I like to make today, and we made in the paper, is that these mental terms, the broad ones such as perception, cognition, emotion, and so on, but even the more focused ones such as attention, memory, decision-making, are what we said, we believe they're epistemically sterile. And the argument that we're going to make is one that is from taking its at root a comparative neuroanatomy perspective, in which we're going to take the organization of the vertebrate brain as a way to understand the kinds of processes that are constrained by this architecture and link back to the way that we typically do neuroscience or psychology. Obviously, this type of approach and this type of argument about the problem with these domains have been discussed for a long time. Many people here in the workshop itself have discussed them, and so Pat Churchland shared some reactions to some of her suggestions in the 80s to when she proposed some of the problems with these mental domains recently as well to many of us on our Twitter discussions. So the argument that we're going to make is that evolutionary pressures molded central nervous system to promote survival. And if we take a deep dive and a careful look at the vertebrate brain neuroarchitecture, we see that is really a place for that supports and allows an incredible amount of signal communication and integration. This general architecture supports a high degree of computational flexibility that enables animals to cope successfully with complex and ever-changing environments. There's a reason that these circuits are this way, and they have a long history in the vertebrate lineage, at the very least in vertebrates, affording this ability to perform flexibly and adaptively in challenging environments, which is what animals face for survival on a daily basis. If we put this together, then we get to the conclusion, or so the claim is, that the vertebrate neuroarchitecture does not respect the boundaries of these standard mental terms that we've been talking about for decades. Neuroscience for maybe a century and a half. So it is the case then that we cannot take a diagram like we often do almost every talk that we go to in a meeting or every book that we read that has something that takes a label, let's say emotion, and points to a place in the brain and either says that emotion is there or heavily depends on this area. And this is the case not even in when you don't have a single arrow, but you have multiple arrows to multiple places. I still think that that's highly problematic. So let's unpack a little bit this argument that we're making. So what we're saying comes from an earlier analysis of again, that we were saying, the architecture of the vertebrate brain, and trying to understand what it implies for the interactions of so-called, not what we call emotion and cognition in quotes. If we take a look at this architecture and consider the general layout in the case of amniotes, birds, reptiles, and mammals, it is a general architecture that offers a tremendous amount of interaction across the neural axis. So it's not something that has some systems that can remain isolated, but instead is one in which there's a macro architecture that links all these levels across the neural axis, including the pallium, the pallium in the case of mammals, we call that cortex, the pallium, subpallium, thalamus, the midbrain and the hindbrain, also integrated with these levels in very strong and important connectional systems that bring these multiple types of information together so that they can be integrated and taken into account in the service of behavior. So to give you an illustration of this type of architecture, let me spend a few minutes in the basal ganglia. This is a well-known system. What might not be as well known is that this cortical subcortical structure of loops is observed in all tetrapods. So it's in all the amniotes plus the amphibians, although it's much less developed and exuberant, I would say, in amphibians. But it's an architecture that, for instance, in mammals, is one that we all know. It starts with these, we think of it as starting, although it's a loop, but it starts with regions in the cortex that funnel down to the basal ganglia, caudate putamen, going back through the thalamus, passing through the pallidum, creating these loops. So one way to conceptualize this, and one that has been popular, was popular for a long time, and it still has quite a bit of popularity, is one in which these systems behave in a largely parallel separate system. So in one paper that I found that illustrates this one view quite clearly, the authors actually explicitly separate a motivational, an associative, and a motor loops in terms of the sectors of cortex that talk to these basal ganglia regions and cycle back to cortex in a way that creates these parallel separate systems. So the question that we've been investigating together with my colleagues is a vertebrate brain like that, or even remotely like that? And so when we take a look across vertebrates, let's start with mammals. What we know about the basal ganglia that is really important to understand is that the different sectors of the pallium, of cortex, they all project to two parts of the basal ganglia, either through ventral loops, let's say the accumbens here, or having also a separate organization of dorsal loops that go via the caudate putamen. So we have these levels, the cortical level, the basal ganglia level, the thalamus that cycle back here. But how are these actually macrosystems interrelated? And so what we see is that obviously at the level of cortex, there's a tremendous amount of interaction between the multiple sectors of the cortex with each other. So there's information that cross flows here, but also with overlapping projection fields from the cortex to the basal ganglia, there's more and more evidence suggesting that these are interdigitated and interacting in much more complex ways than originally thought. So there's quite a bit of confluence at this level too, as well as when the basal ganglia cycles back via the thalamus, via integrating information in the thalamus, as we're going to discuss a little bit later. So what we see here is really a vertebrate property, or at least an amnio property, that is observed not only in mammals, but in birds with these parallel classes of circuits that are far from independent and have multiple pathways or multiple means of interacting. And what I want to now discuss is give some emphasis. I want to emphasize a third kind of loop that is a little bit less emphasized, but we also believe that is a really important macrosystem, connectional macrosystem that is interacting with these other macrosystems that needs to be acknowledged and understood as well. And that one is of the extended amygdala. And so this macrosystem is present here as in mammals, birds, and reptiles, and to some extent a little bit less developed as well as amphibians. So what is this extended amygdala loop? Let's first define the extended amygdala like many people have defined. All Hayden Intro, some of them most clear and I think profound researchers of this idea, investigators of this idea, entertain the idea that the central nucleus of the amygdala and the bed nucleus of the stria terminalis are a unit that can be understood as what they refer to as the extended amygdala. So we know quite a bit about the amygdala. We've seen slides of it all the time, and this one in humans doesn't show very well exactly where the central amygdala is, which is a little bit more where my cursors are, if you can see it, and it also shows very roughly where the bed nucleus of the stria terminalis is, which is a structure that is a little bit less, we're less familiar. We tend to be less familiar with this structure. So in thinking about this extended amygdala loop, it's interesting and important to contrast with sort of like a standard descending control way in which the central amygdala, for instance, is typically understood in a diagram that we've seen many times since the 90s. I think probably one of the first times that this diagram appeared was by Davis in 92, perhaps earlier, in suggesting the amygdala, for instance, engages the periaqueductal gray that allows us to control freezing and other behaviors. But it's really about a set of nuclei that control these effects in a descending fashion. So what we're suggesting is instead to understand this extended amygdala very much like a basal ganglia type loop that exists in a parallel organization in which the cortex or pallium converges, has connections, pathways to the central amygdala, and then to the bed nucleus of the striatum terminalis, which is in this sense a pallidum-like region, and returning through the thalamus to the cortex in mammals. What is really important to understand here is that if we understand this level here of this organization in terms of evolutionarily speaking, this is the cortex and pallium, and an important component of the pallium in the ventral part is the basolateral amygdala itself, which of course we know is strongly connected and interlinked with the central amygdala. So although in this type of loop, the basolateral amygdala has the heaviest set of projections to the part of this loop that cycles back, it is the case that we can see across mammals and birds and amphibians to some extent, almost to a little bit less of an extent in reptiles, I should say, and even a little bit less in amphibians, but definitely in mammals and across birds, that all the different sectors, the lateral, the dorsal, the ventral, and the medial sectors of the pallium project to the central amygdala. And so what we have then is this amygdala loop, this extended amygdala loop, we should say, but it doesn't function in isolation. It is actually a loop that is tied to other loops that we described and have been described for many decades. For instance, the classical ventral loop through the basal ganglia, the ventral striatum or accumbens. And so these loops are intertwined and intercommunicating in very deep ways. So the picture, if we step back now and try to think about the overall picture, what the picture that we want to paint is this of these microsystems spanning multiple levels of the neural axis that actually exist as an organizational principle, but instead of being isolated or parallel systems, they really have to be understood as a system of interlinking or interlocked macrosystems. So the older idea, which still takes hold in many lines of investigation of ways of thinking about the brain, of these systems as parallel loops, really needs to go. Okay, so let me just flip to the second part of the talk. So if we think about it, we have this neural architecture that is allowing all this integration and distribution of signals that the argument is will make any semblance of separate domains that in any way map to perception, attention, memory and learning, language, and so on and so forth, a really poor mapping and a really impoverished way of looking at this overall system. But if that's the case, if not mental functions, then what, right? So one of the questions that I always get when I describe these ideas is, okay, if we let go of these terms, what are we going to use? Are we going to have to redefine everything? And I admit, anyone who proposes such a type of approach will admit, I would imagine, that we do not have at the moment a way of doing this that substitutes this in this sort of neat way, perhaps because of the very reason that this doesn't fit, which is it is actually an artificially simple scheme to understand mental functions and behavior. So one approach that can be used to inspire research, at least, is one that one approach I would suggest is sort of an ecological or ethological approach, in which we try to understand neural circuits and the way they're embedded into these large macro scale systems and in supporting behavior and supporting specific classes of behavior. So how do we do that, right? So I think that that's one direction that I think would be important for neuroscience to develop further and to embrace more strongly. And given the enormous developments in neurotechniques, it's actually quite promising that we are actually going to be going in that direction, given that we can now make recordings with free behaving animals and so on. So it really offers really exciting opportunities to embrace this kind of approach. So let me just very briefly, if you're interested, you can go to the paper to see more details. But let me just discuss one example in which we mention how to utilize, or at least to try to think in terms of these kinds of systems when understanding a certain classes of behavior that are important. So for instance, let's say we take like threat assessment. That is something that has been discussed or threat processing, something that has been discussed extensively in multiple different literatures. So threat assessment, in our perspective, has to be viewed as a highly dynamic process. So for instance, we can take the predator imminence continuum model of Fanslow and Lester and take that as a starting point. But what we would offer is actually that we really need to, even though it was stated in the initial formulation of this class of model and then further developments, that it was meant to be more of a continuum, it was always described in terms of a set of stages that actually follow each other in a more sort of sequential fashion. And we view this as in a more dynamic fashion, in which you might have some sort of general risk perception process that is always going on as the animal navigates its environment. And it's always being guided by the internal state of the animal. Is the animal hurt, or is the animal really well fed and healthy? Obviously taking into account recent and further past learning experiences of the animal and the current context. Is the field all muddy or there's other kinds of obstacles and other environmental context related factors that affect the risk and the perceived risk of the animal. And so we can actually take this as sort of like a dynamic process of going up and down in this axis of perceived risk that also takes into account whether, again, borrowing from the earlier framework that is very inspiring and important, whether for instance, a predator has been detected or has not been detected yet. And so if it hasn't been detected, then the potential prey would tend to avoid locations in the past that perhaps will tend to avoid locations that contain many predators in the past, but that can easily be overwritten in case the internal state indicates that they really need food, they desperately need food at that moment. Anyway, so it's progressing here in this back and forth and along this gradient of risk in a way that takes into account the information in the environment, whether the predator has been detected, hasn't been detected, or has been detected. So how do we go about doing this in many models or many schemes, at least in considering, for instance, the mammalian situation, the mammalian case, in which we actually, to be honest, still know rather little about these processes of threat assessment, and especially if we consider them more as dynamic processes. So one thing that we know from mammals, and building on other vertebrates that have been more thoroughly studied, is that we have a system that heavily relies on close interactions between the periaqueductal gray and the superior colliculus in mammals, or the optic tectum in other vertebrates. And one of the things that are heavily emphasized is kind of this descending kind of processing. So the superior colliculus engages circuits that can move the animal and orient the animal and do all sorts of things. The PAG also can engage all sorts of descending pathways depending on this stage of threat processing, for instance. But what I would like to suggest is that, as in every circuit that we're considering that can be viewed in a more local fashion, we need to understand how it's embedded in other circuits and what this embedding entails in terms of the behavioral repertoire that animals actually exhibit. So just to give you an example of this, the PAG, for instance, projects to a nucleus in the thalamus, the paraventricular nucleus of the thalamus. And this nucleus actually happens to be a major hub connectivity, anatomical connectivity hub that links multiple connectional systems. So what we know is that this thalamic nucleus is actually a key structure in the extended amygdala loop that actually brings the information loops back to cortex. It loops back to pallium structures, in this case, the basolateral amygdala. And it also is connected and links up with the ventral striatum, which is part of the classic basal ganglia loop that we've discussed before. So in effect, bringing together these kinds of loops in a way that situates the behaviors that are ongoing in the context of processing and processes that can be instantiated and supported by these loops. So what I would like to suggest is that instead of looking at dynamic threat processes or threat processes more simply, as has been done historically, even forgetting for a second the more dynamic aspect of it, the more temporal aspect of it, instead of thinking about what is exactly the PhD doing and what exact signals is it sending to the superior colliculus and vice versa, and perhaps one or two other structures here in the midbrain or in the basal forebrain, what impact does the hypothalamus has in modulating these signals. But instead of just focusing on that, which is, I'm not denying its importance, we should really think about how it's embedded in these larger scale circuits. And in fact, it goes even further, because if we think of a structure such as the superior colliculus, there's also very strong anatomical evidence that in mammals, as well as in other vertebrates, it's actually participating in important ways in these important subcortical loops that cycle through the thalamus, striatum, and substantia nigra vitae here, or in cortical paleo loops that loop around in important ways again. So again, the context within which these kinds of processes are taking place, if we're going to have the chance to explain more complex behaviors, needs to be considerably expanded. Okay, so let me just wrap up here, just a final few thoughts. So the accounts that are guided by the standard mental terms, which is dominant in both psychology, but to a great extent also in neuroscience, terms like perception, emotion, cognition, but even more refined terms, sub-aspects of cognition, what's specific type of control and whatnot, they really employ a language, these accounts, that really emphasizes the independence and separation of the mechanisms, like the heavy degree of specialization. The history in a way of neuroscience has one of always seeking this kind of reduction to the exact circuit. But is it really here, or does it really just receive a signal that is related to this other feature type of story? That's the story, the history of neuroscience in one sense. And what we need to promote instead is a language of interaction and integration. the claim, as I just said a minute ago, that this language of interaction and integration is really necessary if we're going to have the ability to elucidate how the brain really supports complex behaviors.
All right, we're looking at about 3 more minutes left.
Yeah, I'm 1 or 2 minutes off, maybe 1 even minute.
Great.
So just a final thought here in terms of how these local and macro circuits assemble. I like to suggest that we have to think about their coalescing across the neural axis in a dynamic fashion that is highly context dependent. And some ideas, some initial thoughts about how this might be happening. If you're interested, I developed some of these ideas quite a few years ago, I guess now in 2017, it came out in 2017 in what I call these functionally integrated systems that again span multiple levels of the neural axis. So a few more details, if you want, you can encounter there, but I definitely need to revisit that and work on version 2.0 if at all possible. So final slide, the mental domains of neuroscience, the neuroscience vocabulary that we already repeated too many times, are too impoverished and problematic conceptual anchors for us to progress. Thank you very much. I'm done.
All right, thanks, Luis. So we're running low on time. I thought I would give preference, therefore, to questions that are more specific to Luis. And there was one question, for example, that there's actually 2 questions that are related, but I'll ask the one first from Anthony Dick. Reliable networks gleaned from resting state fMRI are popular frameworks for investigation, but are they useful? They are not linked to specific behaviors, and from an evolutionary perspective, dynamic networks evolve to solve specific problems, support behaviors. Is it better to start from the problem the organism is solving and then try to understand the network that is recruited, rather than start with the network and see how and then see how it's used in?
Yeah, just very briefly, because obviously this would be a half-hour discussion, right? So I'll just say 30 seconds and we can come back to it later. But you know, I maybe this part of the video you can edit out, because I could get in trouble because I'm involved in fMRI research in my university. But I'm very skeptical that fMRI really is a very useful tool in this respect, because these large-scale systems are interesting, and I think that we need to try to understand what's going on. But I don't think if the example was as looking for them from fMRI, I really don't know exactly how much we can really build from that knowledge. I mean, maybe others disagree, but I'm hugely skeptical, because essentially the view is too static. In a nutshell, it's too static a view of these systems, while I view this as much more dynamically assembling and disassembling, coalescing and dissolving. And in a way that picture is just too slow motion for us to really understand something more important, in my view. And so I do think that we shouldn't start. I don't think we should start from those networks, if there was a question. And I think that we can start from other circuits and for circuit ideas from that itself, but also with anatomy and many other things. But I definitely would actually bring behavior to being an important, complex behaviors, natural behaviors, being an important force in driving the field forward to promote understanding.
That's great. So Lisa had something to add.
I just wanted to emphasize something that Luis said, because I think it's super important. Those intrinsic networks, I worked a lot with those intrinsic networks, and in maybe 10 years ago, I really did think that those networks were performing separate functions, kind of domain general functions, but those networks are an artifact of data analysis.
Okay. Well, so, I didn't want to speak like that, because, you know, I would get
well, I can, because I've published it. I've published it and I've shown it. So they're really an artifact of, so for example, I just showed you this quickly, this image, right, that of 2 of these networks and their overlap, but you always see the networks, you know, shown like puzzle pieces on the brain, as if they're completely distinct in this very kind of modular way. You never see the subcortical extents. And there are all statistical reasons for that. And I guess what I would say is, and I've written about this a little bit because I'm criticizing my own work as much as anybody else's, but it's, you know, from 10 years ago. And that is that when you have certain assumptions, like you think brains have modular systems in them that only have discrete places of interaction, that guides your analysis.
Yeah,
or for example, you know, there's a beautiful paper by Gonzalez Castillo et al. In 2012 in PNAS, and all they did in this paper was increase the within subject sample size.
I remember.
And model the hemodynamic response curve separately for different parts of the brain. And for a very, very simple visual attention task, you don't just, if you only do a standard experiment with like 50 or 100 trials per person, you know, you get a nice frontoparietal control network and the visual system. Yeah. And if you substantially increase the sample size within a subject and you model the hemodynamic response curve, you get something like 90% of the voxels show a change in increase. So I guess my point is that if you use other methods that don't require such harsh thresholding to find signal in noise, you end up recovering the connectome. That was just my point, is that those networks, and I'm saying this as someone who really, you know, in 2006 started to advocate for that view, because I was noticing similarities between resting state networks, like intrinsic networks, and what you would see in tasks. I've really changed my view on that based on the data, and I think it's exactly
that's great.
What you said. I'm not going to ask you to answer anything, but I just want to say you should have a look at Larry Swanson's view of triple descending projections from the cortex to the striatal parts of the brain to the pallial parts of the pallidum, because I'm wondering whether that's not going to get you the kind of parallel integration that you're talking about, but it might get you a vertical integration that he says holds across the entire cortical sheet. And he, not just the parts we're familiar with, and also he calls it mammalian, but right off the bat, the first image that you showed, I wrote down a question like, I wonder if this is a general organizing principle that you can see really even in early vertebrates. It's just something for you to have a look at.
Yeah, no, thanks. Yeah, no.
So we definitely opened up a can of worms, but it's such a lively can. Let's keep it going just a little longer. So we're going to delay the next talk. So
you're sure?
It was a good question. I think it launched. I don't want to stop a discussion. So let's hear from Pat.
Okay, thank you. I'm unmuted. So I was really taken by Luis's worries about the integrity of the folk psychological terms like attention and remembering and so forth. And as you kindly observed, this is what Paul and I used to advocate, actually, under the description eliminative materialism, for which we were roundly beaten up by many people, especially, of course, philosophers. But I think the analogy that I find still rather useful goes something like this, that where we are in the psychological categories is where physicists or chemists were when they said the elements were earth, air, fire, and water. But they didn't yet have a way of talking about the elements as we ultimately got them in the Mendeleev diagram. And we're sort of in that in-between state where we don't want to just say, well, it's integration or it's coordination. We want to be able to say a little bit more than that. And I think a number of people are thinking about what the new categories might actually look like. And one line comes out of Krishna Shenoy's lab at Stanford, and as it happens, Mark Churchland is also doing this, where they're using the structural power of linear algebra to be able to talk about what certain large-scale systems are doing, such as avoiding entanglement and so forth. And it's kind of like what you finally get when it turns out there's hydrogen and oxygen and iron and so forth, as opposed to earth, air, fire, and water. But it is a real dilemma. And I think the important thing is that you see the dilemma, and what you're doing is directing us towards all of the anatomical, physiological features that we've been neglecting in order to kind of preserve the integrity of earth, air, fire, and water.
Right, yeah, and that's very interesting. Again, in the interest of time, just the 10 seconds, and we'll come back to this maybe hopefully later, but I do have one concern, however, that finding a set of basic primitives that essentially kind of span us back with all the mental is sort of an approach that I find problematic. So I think that, finding these elements, these basic elements that when you have like some vector bases that will span the space of all of mentation, I do have some concerns about that view. And so but anyway, that would take us too far afield.
Well, I wouldn't want to say that the analogy goes that far. I think we have no idea what, I think we're really groping for the new categories. And they aren't going to look like how they look in physics. They're not going to look how they look in chemistry. They're going to be unique to biology.
I see, I got it, yeah.
Yeah, so sorry about that. I should have been clear.
No, I understand.
Well, let's come back to that, actually. I think we're going to be coming back to that a lot. But I wanted to get started. So we're starting late already. We're starting 11:22. And the next speaker, I really want Barb to be able to get in here because of all of us, she's the one who's really got the most evolutionary credentials here. So Barb Finlay is a Keenan professor of psychology at Cornell, and she uses developmental and evolutionary approach to study the structure and function of the vertebrate nervous system, particularly the visual system and the cerebral cortex. And the title of her talk is Peaceful Computational Coexistence in the Evolving Forebrain, Separating and Integrating the Olfactory Limbic System and the Neocortex. So Barb, if you could share your screen.
I believe I have, so can you see it?
No.
Okay, let's. There we go, I think that should do it.
Yep, we can see it.
Okay, I'm just going to leave it in the, so are we still good?
Yep, looks great.
Okay.
All right, thanks. Take it away, Barb.
Right, so. Okay, so first, thanks for organizing this thing for Paul and Ben. I think we're in some sort of beginning of a very pleasant explosion in the consideration of real evolutionary neuroscience by a number of disciplines. And I'm going to be talking about a very specific thing here, which is the change in the volume of two parts of the brain. I have pictures here of two that make a maximum contrast, two brains of pretty much the same weight. The animals are wildly different body sizes, the agouti and the owl monkey. And the owl monkey is a cortex specialist, I will say just for the moment. And the agouti shows you a large amount of olfactory bulb and immediate olfactory cortex. And the nature of these two structures has been a problem for a long time now, but although this is a very specific question here, I'm in fact trying to go directly at all these questions about why things are the way they are, as the other speakers have brought up. So why is the brain the way it is? Is it because of its history and descent with modification? Is it because it's regulating the body, as Lisa said? Or is it, or we best understand it by looking at complex behavioral systems, as Luis just mentioned? We've got, in my particular environment, psychophysicists talk about optimization of sensory systems all the time, or opposite motor systems. So where do we go for that why answer? And this particular question pushed me in a direction that I never thought I'd go, because I'm very much of the function, complex behavior first. So let me describe that. So this is a gigantically old persistent puzzle here. And what I'm showing is something essentially from 1973. There's something that Harry Jerrison, who's pointed out here and very clearly in that group photo, that he gave in his MacArthur lecture on the evolution of the human brain. And that's the fact that if you look at how brains scale, vertebrate brains, mammalian brains to start with, you see that the olfactory bulb and its immediate connections acts different from the rest. So here, what I've done, I'm going to be showing a lot of graphs, and usually the point is not the graph in the detail. So to help you parse this stuff, everything that has olfaction in it, like this, will be highlighted with red, or here in this factor analysis diagram, so that you can see just what is being evaluated first. So here I took from a factor analysis that Harry Jerrison did of these large chunks of the brain, the basic telencephalon to myelencephalon, with a few differences. That the first factor you get when you look at those big brain chunks is sort of a whole brain factor, in which neocortex is usually loaded the most. But a second factor that brings up olfactory bulb, and also a few other structures that tend to be associated with it. So I just picked one here. So here's the size of the cerebellum versus size of the brain in a collection of animals, but compared to, with the axes exactly lined up here, how the olfactory bulb looks. And you can see the olfactory bulb is all over the place. Now, I want to mention here something that is of interest, because this is a very comparatively small number of animals compared to what is useful. The animal's phylogeny is all wrong. Insectivores have sort of basically ceased to exist. The prosimians isn't used as a category particularly. The statistics are all wrong. All that stuff, you'd never ever get this published, and nothing about this paper is wrong. Okay, so this fundamental observation of this distinction has held up for those 50 years, and it's bothered me for about 25 of those 50. So the question is, let's explore what's going on. What is the reason for olfactory bulb enlargement? So we need more comparative data, how it changes. So here we're looking at now a much expanded data set. We pretty much still have to use the original anatomical categories in order to use any of the older data, though that's been repaired rather recently. So here I've just put a pink square around the particular relationship between olfactory bulb size and total brain and what you'll see is this: if we now look at not just a collection of mammals, but rather mammals, birds, frogs, salamanders, and sharks and rays, and ray fin fishes, we'll see that the phylogenetic variation in the relative size of the olfactory bulb is also is like the within mammal version of it. So starting from Jerrison and going here, what's the reason for olfactory bulb enlargement? So Jerrison came up with the idea, the concept of proper mass, that the brain allocates the most tissue masked to the most used information source. And he characterized this as animals that were olfactory dominated versus visual dominated one. And also I want to credit here, virtually every slide that you see here has a major collaboration, either from Dick Darlington, Christine Charvet, or both. So I want to make sure I get them on the record here. For those of you who don't usually work with allometry, I just want to point out here, this bottom, that everything is in natural log or log coordinates in the things I show you, so that things that look linear here, when you look at their pictorial volumes, you get a much different impression. So here's from fish to mammals for forebrain in measured volume. Here's how they would actually look to you. The goldfish is invisible, going up through the whale, shark to cat and sheep. So just keep that in mind for the future thing. So we have proper mass, and then so let's look at it some more. We have, along with Kara Yopak and a whole group of people who were doing MRI on as many chondrichthyans, that's sharks and rays, that they could, just to look at the how these animals vary as well. Now we can't, this is using catch, doing MRI and release whenever possible. So this is not the decimation of the shark and ray population, but exploring it. So we've added a great number of sharks and rays to understanding. And so we can separate olfactory bulb from rest of forebrain. And here's a first mapping down here. I direct your attention here first to the factor structure in mammals. And what we've done, we found two factors. The first one is the one that loads on whole brain, and I think particularly here neocortex. So the axes vary here. This is percent of variance, but this variance is 98 percent. What is it? 96.47% of the variance. Then for the red factor, the olfactory one, we see that in mammals it loads on the remaining variance loads on olfactory bulb, olfactory cortices. And then our next set of things are hippocampus, septum, and so forth. And we get the same kind of overall separation where factor 1, the whole brain, factor 2, olfactory bulb, and some parts of telencephalon, which in this case would include olfactory cortex and the like. Now, what's causing this? Let's see. Sorry. My usual method of indicating things or interacting with moving the slide along. Okay, so what we've done here is to just separate the animals into high and low olfactory sizes and then compare it to their environment. So the first thing is general ecology in these. So there's the behavioral ecology. General names would be benthic, which is that the animal was close to the bottom of whatever it is, whether that be a shore or a river or sandy beach or whatever like that, versus pelagic, open water. And then within those categories, we have some special niches which we're picking out. And one of those is reef fish. And the second would be deep water fish. And so we'll see here that, okay, who has which animals end up on the high olfactory side of that? And what we find is it's the ones that are deep water to some extent, and not the animals that are mostly reef, presumably rather sunny environment ones. So it's not a large correlation. The olfactory bulb does not correlate inversely with the telencephalon, but there is a significant but loose correlation between olfactory bulb size and the amount of light in your environment. So some further evidence for a sensory function version of what's going on with the olfactory bulb versus rest of brain. Okay, so this is sort of us going on to investigate within species. This is individual variability using Dieter Kruska's set of who looked at minks and swine or hogs, depending on what they're found in, and males and females. And we have 47 animals all together. And the interest in this is, you have to find variation in your variable of interest at the individual level for it to be considered as a source of deviation to be selected on. So that was our limited motivation in this particular thing, and we were kind of surprised at the strength of the signal we got from the individual variability. So here, this is the same thing you saw before with the mammals, 2 factors, and most of the brain co-varies strongly in the plus 95s. Factor 2, though, the olfactory bulb, well, here we find the same separation in the sharks and the rays with the olfactory bulb showing considerable independent variability. The septum is showing up in this particular data set. But again, this distinction on the olfactory dimension, then Christine looked at a rather limited set of human MRIs, and the principal components vary to some extent. But if you look at the red ones here, what we are picking out again as a separate factor, as a separate component within more than one factor at this time, is hippocampus and olfactory. And I should mention olfactory bulb was never measured in those things. So that's sort of, we're missing our major component of that variability in a lot of primate things. And I can't imagine, I usually can't help myself but to put this last thing on the right in from the individual variability study. The only animals that fail to follow the general vertebrate plan are laboratory mice. So, so what we have done is to put our 47 animals in there, having generated the axes on which they lie on principal component 1 and 2, and 11, we called 11 separate strains of mice, individuals in this case. And what we find is that 3 of our 11 mice are outside of this 95% confidence interval general vertebrate pattern as established by these 47 animals and 1 pig. So just to consider. Okay. Okay, so it's pervasive. This factor is pervasive. It has a possible link to genetics, but we wanted to push it more to get more close to the sort of functionality of the olfactory and limbic factor. And so with Roger Reep, we looked at aquatic mammals, and a large number of aquatic mammals, particularly the manatees, the sirenians that Roger has studied particularly, and seals, and of course whales and dolphins and so forth. The latter 2 we didn't study in this one, but those animals who spend a substantial fraction of their life under the water now basically have very much degenerated or lost their olfactory systems for the simple reason that if you take a big gulp of water, underwater you drown. And no mammal returning to the water has managed to solve this problem to repurpose its olfactory mucosa as yet. So what you see, if you look at those, as we will see in a moment, that their olfactory bulb is much reduced. But also we wanted to sort of enrich the high end. So, and now we're getting to a closer to, current phylogeny here, but we're still using a lot of the Stefan data set and others. But so we added a big dose of Xenartha that would be anteaters and animals like that. And also large carnivores and ungulates for something that was, we're interested in effects of just largeness as well, but we'll get into that here. So again, I've marked in pink the side of the graph that reflects a high olfactory limbic system. And what we have done here is make a ratio of factor to the limbic system as adjusted for the brain core, the rhoencephalon, so forth. So, and this axis is the called isocortex here, the neocortex, norm to the same thing. And what we'll see is a lot is something that did not show up in the shark slide, which is across all these mammals, we have a negative correlation of neocortex and striatum and the factor 2 characters, or the factor 1 factor with the olfactory limbic one. And this is significant both between the different groups that we have here, between the bats, the ungulates, the carnivores, and so forth. As you can see, that each one holds a place on that axis. And also, but within any one of those, we have, just as in the individual data, at the species level, we've got variation on that axis as well. As we'd hoped, we had the aquatic mammals here. Sorry, just can't fix this right now. Is the manatee, there is the black X, and the plus signs are the seals. You see that they are very much in the high isocortex. And then we can see that graphically here. So here we are comparing large carnivores, ungulates, and down at the bottom, you can see the remainder of, and so here for the pinnipeds, the seals, and the manatee, the olfactory bulb is, as expected, quite low compared to the other animals in that same graph. Similarly for the olfactory cortex, that's going to go by a variety of names. So piriform, paleo, olfactory cortex here. So it's also quite low, as you would expect from a structure that has been essentially denervated. But how about the hippocampus? Now, the hippocampus doesn't get a whole lot of direct olfactory input, so you can't really speak of it as being denervated, but nevertheless, people have thought that it might be somehow organized by the olfactory bulb or olfactory information and so forth. But it's lower than you see in the other animals, but it's not that low. And even, and it's retaining about half its volume. So it's not being organized or completely under the domain of sort of supporting input from olfaction. And in fact, if you look at the hippocampus and its size in numerous primates that have either perfectly respectable and functional olfactory bulbs, the hippocampus by far will be the biggest volume component of factor 2. It's not going to be it's going to, the hippocampal volume will be affected by factor 1, the whole brain, and factor 2, but it's very much both dependent and independent on its presence in that category. And what's going on there? So we put up several characterizations in conflict here. And here's a wall of text for you. So that's how I'm phrasing the question. What is this persistent feature that keeps, banging me and maybe others over the head? Why do these structures, the olfaction per se, the olfactory bulb, olfactory cortices, some of the other limbic immediately associated cortices and hippocampus vary so consistently with each other across all the phylogenetic levels we've managed to look at so far. Now, but starting with the notion of proper mass, are we looking at olfaction versus vision? Well, olfactory bulb plus hippocampus plus other cortices versus rest of the brain is not exactly olfaction versus vision. That's way too inclusive a category for that. The hippocampus, this volume component, is only partially associated with olfaction. And as I said, it's the largest contributive volume. So what's going on? Now, I've told you a kind of allometric and evolutionary view of this, but this particular contrast has also interested a number of say evolutionary psychologists and cognitive psychologists. So one here has a lizard brain wants to say separate from the mammalian brain, as Paul MacLean put forth for the triune brain. I just wanted to note that should be included somewhere. The process, it's sensory specialization, but as I just discussed it, one of the major ones that's well known is McClellan, McNaughton, O'Reilly 95, and just recently updated, that this dual structure is related to the kind of memory that each represents. Prevention of catastrophic forgetting by overriding old memories with new ones. So you do that by segregating hippocampal rapid memory organization from slow cortical memory systems. Here's an idea that I had as well, which has to do with lifetime and life histories. So across animals, lifetimes may get longer, but days don't. So that the hippocampus for day-to-day present memories, but you emphasize that your cortex comes big, and it comes big naturally in long-lived species that need long memories because, and I am not implying anything about the causation here necessarily, just the fact that longevity and absolute brain size are very highly correlated. So if you produce a mechanism that makes a large brain, you get a lot of cortex for long memory for free, or the other way around. Okay. And then I can't possibly go into all the other arguments about what the hippocampus per se is for in terms of clapping a functional label on it. But what I was finally pushed to was a computational argument as an answer to the why question here. And that's what I want to talk about. So here's the hypothesis: the primacy of nearest neighbor topographic relationships in the information processing of the thalamocortical system cannot computationally coexist with the distributed property processing of the olfactory and hippocampal systems. And when I say cannot coexist, it goes everywhere from works not quite as perfectly as it should to it can't work at all. We have no idea which. Okay. So what I want to just lay out first is just kind of reiterate and emphasize some of these aspects of the topographic organization of these two forebrain structures. The cortex has been studied exhaustively and interestingly, and it sort of finishes up most recently with the whole set of evaluations of primate cortex done by Markov and Van Essen and Henry Kennedy and a cast of others. But we start from just looking at what a single axon arbor looks like, which in the case of the cortex does not spread forever, but usually has a focus. And in fact, if you look at a number of them, the distribution of axons from this particular site has this exponential fall off feature and a long tail. In the study of connectivity, that's been described and quantified in any number of ways, particularly in graph theoretic ones. And then it proves that this particular distribution is integral, but not the only thing that generates the hierarchy of cortical areas in terms of how much, in this case, how many other cortical areas project to the area. So here we have frontal cortex regions with a great deal of convergence of multiple areas, and occipital cortex at the other side, or basic sensory cortex, which don't get input from so many regions. So as you go more and more frontal in the brain, you become this distribution forces a factor that seems obvious for primary sensory systems, like for vision and audition. Nearest neighbor relationships are maintained because of the nearest neighbor relationships of cells in the retina, which are nearest places in the world, or in the case of the auditory system, nearest neighbors in the cochlea. But this keeps nearest neighbors going in the projections from one cortical region to another, all the way from basic sensory regions up through the most integrating cortical areas. So, and one more thing on that. So here, this is something I did with Ryotaro Uchiyama a while back, looking at how the cortex does this, which came out to be more interesting than I originally expected. So first we take the representation of a monkey brain here, front, back, and they cut it in a couple spots, flattened it out. And so now we have a planar representation of that cortical surface. I stole that with permission and repurposed it with what was known about the sort of the regions of the cortex that projected heavily to each other and had sort of seed zones, which help establish the topography. And I didn't know. And we see the limbic areas, for the most part, are missing from that. Okay, so now what this is take this map and square it up and label it. So why do that? Because I want to represent everything in terms of their embryonic organization. And I want to go back to the original cortical plate and map its sort of descendants onto that. So what we have here is medial, anterior, rostral, lateral. So we have V1 in the back, and we've kept the relative positions and the relative numbers of a monkey in this case. So what you see is two kinds of nearest neighbor topographic mapping that are interestingly sequestered, but not the same as the olfactory limbic one. So the lateral convexity of the cortex has a map of the sort of geography of the body and the outside world. So here we have the earth and the sky, the feet and the head. So what we find across vision, across somatosensory, across motor and so forth, the lower visual field and the lower, the feet and so forth, and doing things with your feet are represented in this area, whereas the top is located, so the sky and the head along this axis here. So a nearest neighbor egocentric map, and a very much an action space here.
Just want to jump in and do a 3-minute warning.
Yep, we're there. Okay, right. And so here, so we have what I've sort of called the temporal ghetto, and sequestered here are a number of sometimes derivations or sometimes independent things that are not so topographic but represent nearest neighbors. So the auditory system is the tonotopic. Cochleotopic is not a egocentric map, but still is nearest neighbor. We have the kinds of parts of face, object and face recognition that are not so egocentric spatial maps. Taste representation and the body interior in insular cortex, which is here. So nearest neighbor topographic maps everywhere, and a big wedge of that is mapping of egocentric space. So so that's how this kind of mapping just dominates the cortex. I don't specialize in, I don't have time to, but what you find something altogether different in the connectivity within the olfactory bulb. So from receptors to olfactory bulb, this is not related to space, and it's sort of that's showing up finally altogether discounted in a number of recent reviews. And then as you go from olfactory bulb to olfactory cortex, very, very early paper that is followed by a number of others showing a really large distribution. Here's Lee and Yuzaki this is just a picture of an axon arbor in CA3 in the hippocampus, just as an example about, and what you find for these two structures, that you find evidence occasionally, greater than chance of patches of local order, but the meso level is very much distributed. So what the argument is here, that what is going on here in the separation of these two systems is a computational problem. So what advantages might, and let's go back for a second just before getting into this. So how do you test that that the information of the kind that you get from smell, or the information that you get from the changing set of scenes or locations you're going through, can't be represented efficiently in the same place? Well, there's a couple hints that might be useful. Like, for example, this same reciprocal relationship of vision and olfaction has even been seen in Drosophila. And I'm not arguing that this has anything to do with representation of a common ancestor, but it might be a common computational challenge that they place. Pat Levitt, years ago, discovered something called limbic-associated marker protein that marks limbic cortices, and he found that people were doing transplant experiments then, and he found that if you transplanted a piece of limbic cortex into the middle parietal cortex, it simply would not make connections with the surrounding cortex. But if you did transplants within lateral cortex, they did just fine. So that transplanted limbic cortex was rejecting all but direct thalamic input and intracortical input. But in adults, that's not the case. That transplanted cortex gets all kinds of projections from lateral cortex. So maybe there's something about the organizational time or something in early development that's critical for this. What we're trying, and this we here is with Nabil Imam, who's now at Georgia Tech, and we're doing some connectionist modeling. And I've just mentioned that we've just started this. So we tried. So basically, what we did was we collected some visual statistics of visual input to use as information, or synthetic olfactory input that have been used in learning studies. And we gave that to a connectionist network, one that was connected full scale, distributed, non-topographic, and a topographic one. And then we presented these and asked the network to recognize either the odors or the letters that had been presented. And here is that incredible success. Well, the vision does better at reconstructing the input if you use a topographic map. And I'm this is the first step. We have so many parameters and challenges of this to explore, but I think you might get a sense of how you might go about looking at this in a modeling fashion. So where we are here. So So
Right,
I
1
was
minute
put, yeah,
left.
I'm put,
Just want to make sure we don't go over and mess up the whole schedule.
yeah, okay, this last sentence, literally last sentence. So, so I'm arguing that there are, it's not a computational constraint, but a computational optimization that's driving the brain in this case. And it's will subsume all the kind of why questions we did before if we understand it from this general computational strategy in which things don't coexist. So, and to finish off, do you not see the epistemological floodgates opening when you focus on the computational challenges of a new ecology? So, that's it. Thank you.
That's a great summary by your so
She's very bright. It's okay.
thanks, Barb, for that. We don't really have a lot of time for Barb herself because we're running out of time.
Okay.
I mean, if maybe we could take one from the, if there's one in the chat, or if somebody wants, if one of the panelists wants to raise their hand. Ben? I just wanted to I don't know if you're familiar with again, I'm mentioning Lucia Jacobs for the second time. She proposed that the olfactory bulb scaling is makes some sense from the context of what is the food source like that an animal pursues. And she's argued that animals that are able to predict, whose prey is predictable, needs to be predicted. So grass is not it, but something that is predictable and whose location is predictable warrants having a larger olfactory bulb and limbic system, whereas animals that like eat grass or just detect prey don't need it so much. So it's kind of an olfaction navigation hypothesis.
Yeah, well, I don't know. I mean, a lot of monkeys eat mostly the New World monkeys that I'm the most familiar with eat all kinds of things, and I don't know, they have relatively small olfactory systems, but I don't see any reason why they couldn't use that. I mean, I can take any set of, any order of herbivores here and pick animals that don't demonstrate that distinction. So, but I'm not going to rule that out. I mean, one of my whole points here is that you have to, scientists and people are not happy with partial correlations, but we got to be. So it might account for some of the address. Some of
biology is messy, yeah.
Yeah. So,
okay, maybe we can get started with the open discussion. I just want to thank Barb again. Thanks a lot. We'll start with the open discussion, and we'll keep it going for. I think we should try to keep it going for at least 20 minutes because there's a lot of good points. And we're gonna maybe bring a couple of people on stage, and we'll start. Actually, we'll start with Joe. Joe's got a comment to raise, so please go ahead.
Okay, so yeah, I have a question for Lisa. Lisa, a lot of your theory and emphasis has been on valence. But when you talked about reward as a brain-wide kind of state, I wonder if you would think of valence that way as well now. Yeah,
so I think, I think my career, you could say, is, I was always kind of questioning psychological categories or Western categories, I should say, too. It's not just that they're common sense, they're Western common sense, they're incredibly ethnocentric. But valence does seem to be a feature that does generalize across cultures as far as we can tell by any set of measures. But I guess what I would say is more like this, that there's all this research on behavioral state in animals showing that it's a brain-wide representation. And I guess the way I think about it is that brains don't detect things in the world or in the body, they're computing features. And some of the features that they're computing are very close to the sense data that are received from the sensory surfaces of the body. And some, as Barb showed us nicely, and it's been very well established now, that there's this compression gradient where there's this multimodal integration of information as you move towards the front of the brain. And even though in the Markov study, they didn't really include cortical limbic areas, I actually think we might want to not call them that anymore because they don't show up on Barb's limbic factor. And we have to stop using that word, because not just because there's no limbic system, as you and others have pointed out, Joe, but it actually, those cortical areas don't show up on that factor. So we have to think about what that means. But more importantly, to answer your question, what I would say is, as you move to the front of brain, the brain is computing more compressed multimodal factors, multimodal features, or we could call them abstractions, multimodal abstractions. And that's what valence is. That's what threat is. That's what reward is. And the reason why that's the case is threat or reward or valence, like positivity, negativity, is associated with multiple different patterns of low-level sensory motor features. And that's the definition of a multimodal abstraction. So I think the way I used to talk about this is that before maybe 15 years ago, is that there was a neural workspace for valence, a neural workspace for arousal. But really what I was trying to get to was this idea that a feature like negativity or positivity can map to, be associated with, come from very different low-level sensory and motor features. And so I would put it in the same class as reward, as value. These are abstractions, multimodal abstractions that a human brain can compute. And I think the fact that we do this so easily and effortlessly as scientists, we should be concerned about it in the following way. And this brings me to something Luis said, and really in response to, this conversation that he was having with Pat, and that is that, the human brain is equipped. So you could say any mammalian brain has this compression gradient, okay? And it's probably there in other vertebrates too, but I'm just going to stick with mammals. But just by virtue of the normal scaling properties, humans have this capacity to do much more abstraction, much more compression than other animals do. And that means that we can look at a fly that doesn't move, and a rat that runs away, and a human who makes a wide-eyed gasping stare, and we can say, those are all threat. But the question is, does that, that behavioral capacity that we see similarity in those three, because the feature of similarity there is an abstract feature that our brains can compute, but does that actually correspond to what those animals' brains can compute, or what those animals, is that actually the proper way to be describing the behaviors of those animals? And that is something I think we just haven't really recognized. So I'm even questioning my own use of valence or, threat or value, because I think we have to recognize that human brains can do much more compression, therefore much more abstraction. We can see similarities in many different sensory motor patterns. We can call many different things negativity. And that means that any kind of functionalist approach to understanding brain function, maybe there's a risk there. That we are imposing imposing features that may not actually describe what's going on in those whatever those animals' brains are.
Yeah, that's very clear, because it wasn't exactly the way I was thinking of the way you thought about valence. I don't want to go into more detail because it's probably taking up too much time. But
I will just say one thing, though. I'll just say one thing that I think this is true for all of us, maybe, and that is that all of us here, I think, are marked by the fact that our way of thinking about things changes with data. Like we're all grappling with data, right? But what that means is we have to be really careful about how we cite each other, because if somebody goes back and, I'm sure this happens to you, it happens to me all the time. People will cite something I wrote in 2006 or 2012 Well, that was a long time ago.
That was a different you. That's right.
And my thinking has really been shaped by, you know, lots of data that either that's new or that I wasn't aware of at that time.
Right.
And yeah, so that's the sort of caveat I would sort of throw out
It's
there.
definitely a problem, but let's go on.
Okay, so I invited Robert Barton just to be a panelist here for the discussion because he had a couple of questions during the previous talks that I think were actually kind of relevant for all of them. So Robert, if you'd want to maybe raise some of those.
Hi, thanks very much. Can you hear me okay?
Yep.
Okay, well, my first question was originally to Lisa, but I think it probably applies to Luis equally, if in slightly different ways. And for a long time, I've been kind of bothered by the obsession of comparative psychology and indeed comparative neuroscience with sort of identifying the areas of the brain associated with higher cognition that have kind of expanded most during human evolution, and sort of thinking that, you know, there are some individual areas in cortex that are really where all the interesting action has been. And so my question to Lisa was that, you know, with this kind of embodied perspective that you have with which I'm very sympathetic, an implication is that we should be focusing much more on distributed networks and their modifications in service of specialized bodies, and perhaps less on single areas that we tend to, you know, think of as being associated with so-called advanced cognition. And I'm not even sure that we should use the term advanced cognition. I'm not sure that it really maps onto anything, you know, going back to the sorts of things that Luis was saying, it really maps onto anything biologically terribly meaningful. And then just to sort of focus the question a bit more, so what people tend to do is to search for similarities, cognitive similarities between very, very different sorts of organisms in the search for, you know, things like theory of mind, and ask whether, you know, apes, crows, dolphins, octopuses, organisms with very, very different bodies who presumably, you know, the nervous system evolution is completely different in those species. By looking for the similarities, if you like, by looking for the signature of what we consider to be higher cognition across those species, we're missing, you know, a vital part of the story. And that we could be asking much more interesting questions, actually, about embodied cognitive evolution.
Yeah. So I'll just jump in quickly and say, I completely agree with you. I would say that, you know, one of the problems with using folk categories is that we put ourselves at the top of a phylogenetic scale, and we're like, you know, what's higher cognition? It's whatever humans do. And so we end up looking at other animals like inferior humans, you know, and that's really a problem because there are animals that can do pretty miraculous things that are very suited to their own bodies and their own, you know, ecological niches. That's also true across development after birth, I might add, that, you know, the way that you look at a newborn brain or an infant brain is not necessarily the same as the way you might look, like, I mean, in terms of cognition or these kind of like abstract categories, because their bodies are different. And I think the extent to which your brain wires itself to the realities of your own body, I found it like very cool and surprising, and just things I never really thought of before. Like, you know, your visual system is wired to the distance between your eyes in your skull. And, you know, your auditory system is wired to the particular shape of your ear. So the sensory surfaces, which leads me to say that the brain is not running a model of the world. It's running a model of the body, including the sensory surfaces of the body, which is how it knows the world. And so the body becomes very relevant. The other thing I'll say, though, just really quickly, is that I think that, you know, I loved Luis's talk. You know, I've written some of those same things, but he's taken it to, I think, another level. And particularly his focus on language is really important. So even those of us, even Dan Schachter, who discovered, that memory is constructed, talks about the storage of memory and the retrieval of memory. And memory obviously isn't stored and it's not retrieved in any animal, including us. And so we don't really have an embodied language, really, Robert, because we largely ignore our bodies in everyday life. We're very, very focused on the world. So I would just sort of say that I think there's a deep problem here in that scientists tend to focus on their own experience as a jumping off point for, and they believe that some scientists believe that, when they see a rat not move or a fly not move, that they really literally are observing a fear behavior. They're not aware of, what they're adding to the observation. So I think we have to be really, really careful of that. And I think Barb's approach is really the right approach, actually, which is to talk about computation. So for example, it's not just like, so in terms of compression gradients, again, you would look at the structure of the olfactory bulb. The compression is happening right at the sensory surface. And that's also true a little bit about, sensory systems inside the body. They are widely distributed, and they are also, the compression is happening, some of it actually, as the signals move up the vagus and A delta fibers. So I think that, the part of the ecology of an animal is its body, literally what the parts. And I think you're exactly right. Yeah.
So Louise, maybe, and Pat also wanted to comment on that.
Oh, just briefly, I was just going to mention that I strongly agree with you, and I think that your sense of puzzlement is echoed by a lot of people. I'm also very puzzled about this almost exclusive focus on some magic parts of the brain that all of a sudden brought the light to human beings, and I think that I don't find that way of thinking inspiring at all, and I think that's just.
Are you deliberately using religious language there, or was that just a turn of phrase?
No, I wasn't. Was I using deliberate religious language? I didn't mean to, but maybe. What I guess what I'm saying is that this progression toward perfection, again now in the religious sense too, is dominated neuroscience thinking to such an extent that we keep on going back to these same things of trying to find a solution in the ultimate neuronal cell type, or the neuronal connection, or the neuronal this or that will bring about these changes, when we have these highly complex distributed systems that have modular components, that have modular structurally speaking and genetically speaking components that have been studied for decades, and that go along 500 million line of evolution at the very least, that I don't think that this is integrated in any reasonable way at all. So I find a lot of the takes in neuroscience heavily uninspiring. And I think that the way you phrase things, and we've been in other venues together, and what I've read and heard you say is, I think that is to me, I find it very inspiring to think about, and it almost like flipping the problem around.
Pat wanted to say something.
Yeah, I just wanted to ask Barb a quick question, and it's this. You said that longevity and brain size are highly correlated. Why is that?
Well, I did avoid making a claim about the directionality of that. Let me give you a couple. Let me give you a functional in the world explanation. Well, the longevity and brain size are also highly related to range. And so, for example, in primates, frugivory is associated with brain size, and also requires memory of highly distributed, non-constant food sources. So you have to remember things over a longer amount of time. So you could go to an explanation like that, which is in the environment. It just turns out that in mammals, you have very little options about how to make a brain larger. You have to take more time for the most part.
I see.
And so if you've got an infant that is taking a long time to make the brain and you want to help that infant survive, you have to at least add that duration of development to your own lifespan, or even more, as in Kristen Hawkes's grandmother theory, that you have an actual non-reproductive period that is devoted to helping extended offspring. So I intermixed a developmental constraint with a functional argument there, which is where to locate the why, and that is the problem that we all have. Okay.
Okay. Thank you.
Mm-hmm.
Okay, maybe we can get a question from the questions that people have posted. I'll just go right ahead with the one that got the most votes, which was by Ash Singh, asking what are advantages of innate emotions from an evolutionary perspective. I guess many of you have opinions on emotions.
I don't think that's a meaningful question. Sorry, Ash, but that's the reason why I didn't answer it.
Because they think innate emotions is just not a category.
I don't know. I don't know what that means actually anymore. So, here's what I would say just off the top of my head. I really dislike this question because I think it's a faculty psychology question, right? It's just pure and simple, not a useful question to ask, unfortunately. So think about, I think Barb made this point, and maybe Luis made, but oh no, I think it was Ben made this point, and also Barb made this point, right? That variation is required for selection. This is Darwin's, also a big point of Darwin, obviously. So let's think about what it means to have something be innate. Something like Hox genes, and now I'm saying something everybody knows, but this is just my sort of simple-minded way of thinking about it. Something like Hox genes, which have been, don't quote me on the time, but what, 500 million years they've been fixed, basically, but which means that they're under very serious selection. This is something I learned originally from Barb, and it made a lot of sense to me that those genes don't change. Because all the animals where there are mutations die and don't reproduce. So they're fixed in a sense by very heavy selection pressures, right? Okay. So anything that we talk about being preserved, of having something be innate, whether it's innate in all animals or innate in only vertebrates or innate in only mammals or whatever, we have to have a benchmark of what that means when we're talking about innate in the usual way that people mean it, which is to say prescribed by genes that are carried over that pass information from generation to generation with no change or very little change. So I guess I don't think there are, first of all, I don't think there are any emotion circuits in the brain. I think that emotions are constructed events in the same way that cognitions are and whatever. Those are the way that those events. So I would say that, cognition, emotion, perception, these are part of a Western theory of mind. They are events that are dictated by the experience that we have with in a particular cultural niche. And so they're not causal in any way. They're not evolved in any way. Evolution doesn't act on these, but there is sort of cultural transmission of, cultural inheritance of concepts and knowledge. And that's why, we experience, anger in a variety of ways, or we might, see anger in a variety of actions, because our brains can construct that using I didn't at all talk about compression gradients and these features and what that the brain, you can think of the brain as a category constructor. That's really what, prediction is. That's what memory is. You can think about it that way. You can think about it in terms of continuous category construction. And the features of equivalence, some of them are not in the world, they're learned. Like maybe not valence, that might actually be too tied to interoception, but some of the features are learned. And that means that they're a way of making sense that your brain has learned, probably through cultural inheritance. So if the question is, what's the evolutionary significance of emotion, that's a really different question than what is the value of innate emotions, if you understand my point.
I think so. I think I feel the same way. Robert, maybe you can ask your question, but I just want to say that we've already cut into the break. So if people want to pop out and come back in, I do want us to restart at 12:35 on the dot with Joe LeDoux. So the more we chat, the less time you have to take a break and grab a coffee. So Robert.
9 minutes.
Thanks. I just wanted to jump in there to pick up what was just being discussed, and because and I want to ask Lisa about valence, and I'm trying to grapple with what she really thinks about valence based on what she said. It's bothered me for a little while that valence might be simply a culturally constructed value judgment, but I'm not sure if that's what you're saying.
No, that's not what I'm saying, actually. So what I'm saying is that I don't think anybody who's ever suffered from real distress, like real visceral distress, where you feel like your insides are being eaten alive, I don't think that that's a cultural construction.
No.
So but I would say that for a long time, Robert, I think that there's been a confusion about, this is something I actually dealt with in my dissertation, believe it or not. It was a really long time ago. The normative meaning of valence and the hedonic descriptive meaning of valence. So no, what I'm saying is that there are lots of ways to feel bad. There are lots of ways where things feel unpleasant. What is a feeling of unpleasantness? Well, I can tell you, after having spinal surgery in the last year, there are many ways to feel. There are many different sets of features that correspond to feeling unpleasant. And that's what I mean. I mean that the sensory motor, the low level sensory motor features that are a part of an episode, an event of unpleasantness vary. So unpleasantness is an abstraction. It's a multimodal abstraction. That doesn't mean it's not real.
Yeah.
It means it's a feature. It could be a feature of consciousness, like brightness, right? Or that's what I'm.
Can I interrupt here? I kind of wanted to hear the whole question that Robert was going to ask.
Oh, I thought that was the whole question. I'm sorry. Apology.
It kind of was, I guess. What else might I think that there are some aspects of affect that are very difficult to put on that continuum of valence. It's not obvious where that certain things like, even anger is generally construed as a negative aspect of affect, and I struggle to see that that's necessarily biologically meaningful. So I guess what I'm saying is, surely there's more sort of biological structure to affect than that two-dimensional model of arousal and valence.
I don't. Well, are you done?
Can you let him finish?
No, that's fine. I
love to hear what you have to say, but I kind of want to hear what else he had to say on the topic.
No, I think that's it. That's fine.
Okay, fine.
Go ahead, Lisa.
Those two dimensions don't well, first of all, I don't really work in that space anymore, but those two dimensions don't represent anything about biological structure. They're features of experience. So my views on this really differ from, let's say, somebody like Jim Russell, like where, he and I were very, similar at maybe 15 years ago. And I would say that valence is a feature, but it can, there are instances of anger which feel pleasant. There are instances of anger which feel unpleasant. There are instances of fear which feel pleasant. There are instances of fear which feel unpleasant. The placement of those words in that two-dimensional space are really the stereotypes of the concepts in a particular culture. So I don't really think I think that space is useful for describing two features of experience, like arousal, for example. It's describing subjective arousal. It's not just do you feel worked up? Do you feel calm? Or it could be, do you feel attentive or do you feel inattentive? Or it could be, do you feel energetic or do you feel not very energetic? It doesn't really specify any of the 15 or 20 different processes going on inside your brain and your body that could contribute to a feeling of attention or feeling of arousal, being worked up, or feeling of having energy.
Do you think that space could be maybe sort of anchored to biology better, like saying something like, there's a dimension for sort of, neurogenergic input from locus ceruleus or something, some kind of states or axes that are actually defined by specific circuits that we can study, and then map the states onto that.
I think I think they're abstractions. It's a low dimensional. It's low in dimensionality because it is abstracting away from all of that high dimensional state space in which arousal or valence probably lives.
But if the axes were actually linked to specific circuits, then they're concrete, not abstract, no?
Well, I think that people have tried that, but they've done it in the way that many of us object to, and that Luiz, I think, very nicely described. So they want to say, that dopamine is arousal, and that, opiates or serotonin actually are valence. And that's just not defensible empirically. So I'm not saying that the two dimensions are not useful as a descriptive space for low dimensional properties of experience. I'm saying they don't map cleanly to emotion categories. They don't map cleanly to any biology. And I could make an argument for, dopamine being related to lots of things, not just arousal. It's a metabolic regulator. Serotonin evolved as a metabolic regulator. So did actually every hormone that we have. So you could always think about it just from that non-psychological perspective. So I think it's a conceptual tool, that two-dimensional space, and we should be using it where it's useful, but not mistake it for a useful biological summary. That I think that whole line of debate is just, what'd you call it, epistemologically bankrupt or whatever.
Well, then let's let Luiz finish it up. All right, so Luiz, we have like a minute and a half, and then.
Well, I was going to agree completely with Lisa. I mean, the degree of abstraction that has gone all the way into those things. I feel this way today or that, whatever. Last morning I felt that other way. It's just the degree of abstraction is so extreme that it's, like you said, transplanting it to some mapping. It's not to say that it's completely arbitrary. It doesn't map like randomly to what the body can generate, but like what are the systems, like one neurotransmitter system versus another system. The way the example that you gave, it's expecting a regularity in something so low dimensional and simple that's the problem. I think the problem is that the mappings are never simple and one-to-one, and we would love it to be, because neuroscience would be much easier and we'd make a lot more progress, but it's just not the case. So if you take any one of those variables, whatever it is, choose your favorite neurotransmitter system, it's just not going to work.
All right, that's great. We're gonna have to move on. So we have like a 15-second break for everyone who wants to, I don't know, breathe. And I'll pass it over to Ben, who's going to moderate the next session. All right.
All right. See you all in 10 seconds.
I've got to go and catch a train. So sorry, I have to leave, but I'll watch the recordings later.
Okay, welcome back from that extremely brief break. Thank you very much for attending this. We're doing really great. I'm really excited about the quality and quantity of the engagement that we're getting and the questions, and obviously the talks are fantastic. In order to keep things on track, I'm going to introduce our next speaker very briefly. Joe LeDoux is the Henry and Lucy Moses Professor of Science at New York University, and he is the director of the Emotional Brain Institute. His research is primarily focused on survival circuits, including their impacts on emotion, such as fear and anxiety. Take it away.
Thanks. So let me just share my screen and get started. All right. So I've spent most of my scientific, here, let's start with this. We have the first 3 billion years of life on the top and the last billion on the bottom. The top part is all unicellular and the bottom part includes multicellular as well. Now, I spent most of my research career studying what role that, what we could learn from mammals about human behavior, and especially in terms of learning and synaptic plasticity. And at some point, it became apparent to me through the work that was being done in invertebrates that there was something going on down there that was directly relevant as well. It wasn't just to me, but to the entire field. And as a result of that realization, at least in my mind, I ended up going back to the beginning of life in this book that came out in 2019, The Deep History of Our Cells, the 4 billion year story of how we got conscious brains. So that's what I really want to tell you about today. The title is From the Way We Were to the Way We Are, and let's jump into it. So I did, been working on rats and threat processing, or what we used to call fear conditioning, for 40 years or so. Simple explanation of what this is a sound is presented to a rat. If we'll ignore it after a couple of times, but if the sound is paired with a brief and mild shock, the rat will then freeze the next time it hears the tone, and will continue to freeze throughout its lifetime, and for the most part. The threat goes into the brain through a sensory system, is processed in the brain, and out come these behavioral and physiological responses like freezing, as well as changes in blood pressure and heart rate, and various kinds of stress hormones are released in the process. And by connecting the dots in the brain between the sensory system and the behavior, we were able to come up with a structure that was very important. I'd done my PhD work studying split brain patients, where what you begin to think about is how information goes from area A to area B, or from hemisphere A to B in that case. And at the same time, this work on invertebrates was being conducted by Kandel and others that showed how important it was to trace the flow of sensory information through the nervous system to understand the sensory motor integration. And this approach took me and my colleagues to the amygdala. And from there, a lot was discovered. So we learned a lot about the behavior. I mean, we, the field, not me in particular, but although I contributed a lot to this, what do we know about the behavior? All the behavioral principles were known from behavioral psychologists studying Pavlovian conditioning, but what we contributed was the circuitry. And you see a lot of information here about the amygdala integrating different kinds of inputs and expressing responses through various arousal systems, as well as through behavioral control systems, autonomic systems, and hormonal systems. And of course, we learned a lot about the cellular molecular mechanisms as well. And that's where I want to turn to for a second here. So if we look at this panel here, what we see is on the left side, a presynaptic neuron, and on the right side, a postsynaptic neuron. David Glanzer at UCLA put together a summary of all of the molecules that had been implicated in synaptic plasticity in pre and postsynaptic neurons, both in invertebrates and invertebrates, and in particular mammals. So everything you see there exists in flies and bees and worms, as well as in rats and mice and other mammals that you might want to study. So what's the likely explanation of how mammals and, let's look at here we go, vertebrates on this side of the tree of life top here, and invertebrates over here. How did they get these same molecules to be involved in synaptic plasticity? Well, the likely answer is there was a common ancestor that existed about 635 million years ago, according to the genetic analyses, that passed this on. And so studies have been done showing that all these synaptic plasticity, ignore the thing in the middle here, that all the synaptic plasticity molecules that we've been looking at are present through vertebrates, invertebrates, lower metazoa, and also in protozoa, the single cell ancestors of animals. And why that's interesting is they don't have synapses. It's not that they have all of these synaptic plasticity molecules, but they have significant ones. For example, NMDA receptors, the golden fleece of synaptic plasticity. They have components of the NMDA receptor. So what are they doing with this if they don't have a nervous system? Well, obviously the answer is that animals took what they got from these single cell protozoa and modified it to create synaptic plasticity in neurons. It's not like the single cell used these same molecules necessarily for synaptic plasticity, but maybe because despite not having a nervous system, protozoa and bacteria both behave. They approach attractants and they avoid nutrients. So they even learn. So evidence in protozoa, classical conditioning, for example, bacteria and so forth. So the idea is that behavior and learning does not require a nervous system, and it may be as old as life itself. Only animals have nervous systems, but perhaps all life forms have to have some way of detecting and responding to danger and doing other things to stay alive. So the implication is that behavior didn't arise for the psychological reasons that we impose on our understanding of behavior. And I think that's a great point that Luis and I think Paul will make later, that these psychological categories are things that we've invented, and whether we've got them right, maybe or maybe not. But how could, when these ideas were invented, thousands and thousands of years ago, in some cases, how could they have possibly got it all exactly right? But the point is that these psychological that behavior itself is not part of the psychology we have so much as part of our organismic toolkit, survival kit. So what are the requirements of a cell? Well, it's got to be physiologically viable. It's got to make energy, balance fluids and ions, withdraw from harmful agents, among other things. And it has to reproduce for the species to survive. But the same is true of an animal, including us. These are basic requirements that we have that sustain our life. Now, there's a difference, though, between survival requirements and survival implementations. The requirements are things that all animals, or all organisms in this case, must satisfy to survive, whereas the tactics or implementations are the way that each species has come up with a unique plan, a unique solution that's tied to its body plan. For example, defense against harm, all animals do that, but some freeze, flee, or fight. That is, run, fly, swim. Even within mammals, you can see that some run, some fly, some swim, some climb, depending on the body plan that they've evolved. Energy management. Some organisms digest internally, others externally. Others use photosynthesis for processing energy. Fluid and ion balance. Some animals drink, others absorb. And reproduction, there's asexual and sexual. So depending on the body you have, which is evolution's solution to survival, those are the kinds of implementations that you use to defend, manage energy, balance fluids, and reproduce. So here's some examples of you have one animal that's going to climb up, another is going to fly away, another is going to swim away, another is going to run away. And so it's not behavior that's conserved in evolution across the large space of evolution, but survival requirements. You have to be able to escape from harm, but the way you do it is dictated by the body you have. So when we talk about these things, we humans, we tend to impose our experiences on the interpretations of these. So here we have defense, energy management, fluid, and ion balance, reproduction. When we have these, we feel fear, hunger, thirst, and pleasure. But when we talk like this, we're conflating our survival, we're conflating our emotions with the long history of these survival responses. Again, it's not the responses themselves, but the survival functions that have been enabled by this long history. So in a sense, you could say that the amygdala in us and other vertebrates is a kind of endpoint or a current endpoint of these long sets of changes that have gone through all of these kinds of animals that allows them to survive. Again, it's not that we've inherited our amygdala or even a neural circuit from bacteria, but instead, perhaps what we've inherited is this need to defend ourselves in order to keep the organism alive. So all organisms detect and respond to danger. All metazoans have a circuitry that controls responses to danger, and all vertebrates have defensive circuitry that depend on the amygdala and related areas in the hypothalamus and periaqueductal gray that we've heard about already. So the amygdala, by studying these behavioral and physiological responses elicited by threats, the amygdala became a fear center. And I'd often be introduced in lectures for decades as someone who had discovered how the feeling of fear is represented in the amygdala, which is not something I ever really said. Or maybe like, as Lisa pointed out, you said something in 1996 and, you're held to be accountable for it today. But for the most part of my career, even back in 1996, when I wrote The Emotional Brain, I was emphasizing that emotions are cognitive interpretations of situations, including fear. But I wasn't always so clean with the language, and so I could be accused of having some sentence somewhere that says that the amygdala generates fear itself, but I don't think you'll find many of those. So the idea that threat activates the amygdala, fear is aroused there, and as a result, fear responses occur. In a way, this is based on an argument by analogy with human behavior, which I think we have to credit Darwin or discredit him for. We can credit him for giving us this, but I think it's something that he shouldn't have given us such a strong opinion about. For example, we often freeze and feel fear when we freeze. Amygdala we often feel fear when we freeze. Amygdala circuits control freezing. Therefore, we inherited our amygdala from our mammalian ancestors, and they must feel fear when they freeze. So Darwin was very prone to proposing that, for example, emotions are mental states that we've inherited from animals. But I think there's another way to think about this. Well, I've already kind of said this. We shouldn't be projecting our experiences back onto other mammals. That's usually where the emotions stop, but not necessarily, as we'll see. So, anthropomorphism has been proposed to be an innate part of human nature, something that was beneficial to our early ancestors in their quest to survive by kind of treating animals as little people so that they could get into their psychology in a sense and try to get them to do things for example, agriculture depended on the domestication of animals for supporting the work. So even though it's innate and was useful to survival, that doesn't mean it's a valid construct psychologically. So, in coming to terms with fear, this sense that I had over the decades was that at first it didn't bother me that I was being associated with the amygdala as a fear center in the literal sense of the word fear. But by 2012, I sort of had it. And so I wrote Rethinking the Emotional Brain. The idea here was that the threat doesn't activate fear, which causes the behavior. It activates a defensive survival circuit, which produces a defensive behavior, not a fear behavior. So, neuroscientists can we like to describe things in terms of systems, cells, synapses, molecules. And I think this works equally well in all animals, vertebrates and invertebrates. There's no need to call upon such concepts as fear. We can explain everything about why a tone elicits freezing in a rat on the basis of Hebbian plasticity, where the CS and US have been integrated in neurons, in part in the amygdala. The CS then activates those neurons and produces the freezing response. If during the learning there was a squirt of norepinephrine from the locus coeruleus into the amygdala while that plasticity was taking place, the strengthening of the synapses occurred much stronger. Or even during the retrieval of the memory, Lisa doesn't like the word retrieval, but the expression of the behavioral response in response to the stimulus, if norepinephrine is presented, that will cause a kind of storage, reconsolidation in a stronger way. We don't need fear in any of this. We just simply need cells, synapses, and molecules. So this doesn't mean fear is an irrelevant construct. It's just not what causes innate responses elicited by conditioned threats in either humans or animals. So, why does it matter? Well, I'm not the first person to question this kind of stuff. So Francis Bacon said scientists should be vigilant and guard against tacitly granting reality to things simply because we have words for them. Mandler and Kesson in '64 in a very good book called Language of Psychology said the common language is full of quasi-psychological assertions, and the language in which these are framed is inadequate. Jack Block, a personality psychologist at the end of his career, psychologists have tended to be sloppy with words. Summary labels and shorthands control the way we think. And Melvin Marx, there's a semantic danger that results when a common language term is used as a scientific name for an intervening variable or a hypothetical construct. The variable or construct becomes infected with the subjective properties of the common meaning. And that's what has happened in the case of fear, for sure. So, in genetics, it doesn't matter if we give things cute names like hedgehog. Nobody believes there's a hedgehog living in the gene, or that the hedgehog is making of course, the hedgehog may make the gene, but it has no relationship. No one is like, misinterpreting what the genetics mean by giving it a name like hedgehog. But when we come to psychology and neuroscience, giving words, common language words, especially mental state words, using them to account for behavior conflates the difference that I talked about earlier. The history of survival with emotions, which I think are probably much more recent, not necessarily just human, but certainly more recent than the history of danger. So here's some examples of how we get in trouble with language. So I'm from Louisiana, where we say crawfish. So we say crawfish treated with a benzodiazepine were more exploratory, that is less inhibited, were a chamber where they receive shock. 2014 in Science, not sure it should have been in Science, but it's a fine paper. But here come the headlines. Science, anxious crawfish can be treated like humans. New York Times, even crawfish get anxious. BBC, a little more tempered, crawfish may experience a form of anxiety. Another example, David Anderson, one of the most prominent neurobiologists in the United States, maybe the world, is interested in flies and their freezing behavior. And he said an emotional state, possibly analogous to fear in mammals, lies between the threat and the freezing behavior in flies. And by studying flies, we can learn important things about human emotions. Maybe so, but that depends on what we mean by emotions. And you can see here in the headlines that what the public thinks about emotions is different from what Anderson was trying to say. Flies have feelings, fear, and maybe more. Flies experience emotions like fear and might offer insights in how the brain makes feelings. So a practical example is for this and the problems that result is anxiolytic drug development. Since the 60s, lots and lots of money and lots of animals have gone into the search for new and better medications. But by 2010, after decades of research, the CEO of GlaxoSmithKline concluded that the effort had failed and new investments wouldn't be made because of the low probability of success. Andrew Holmes, a leading researcher, said something similar. Novel results and novel agents in rodent studies have very rarely translated into effectiveness in humans. So, this has resulted in a scenario where a psychiatrist will tell you the medication will turn off your fear center and you'll be able to go to a party and not feel afraid or anxious. But there's another scenario, which is the physical symptom scenario, which is that the medication, this would be a more, I think, appropriate thing for a psychiatrist to say. The medication affects systems that control behavioral and physiological responses in challenging situations. You'll find it easier to go to the party, be less avoidant, and will feel less jittery or less aroused. While you will probably still feel anxious, you'll be better able to cope with the threatening situation and can use it as a way to become physically more comfortable being there. This is not a cure, but it might help you cope with your symptoms. So unfortunately, this is the way the drug companies have promoted the medications and how the psychiatrists have interpreted them. And it's no mystery as to why people aren't more satisfied and why they don't seem to be working better. I think the behavioral symptom approach might have been better. So we need a conception of fear and anxiety that recognizes the importance of innate circuits that have been inherited from animals, but that doesn't confuse those circuits with circuits that underlie conscious feelings of fear or anxiety. So the drugs they're developed in psychopharmaceutical industry settings to work on these survival circuits. You're measuring behaviors like freezing and avoidance to get a drug that is going to make the person feel less fearful or anxious. But if people are feeling less fearful and anxious because they're cognitively interpreting the situation in which they find themselves, then it's not a surprise why the drugs are not more effective. So in 1978, Mike Gazzaniga and I wrote this book based on my dissertation. And we said emotions are conscious experiences that result from cognitive interpretations of in situations in which we find ourselves, including our behavioral response. So that's why I turned to rats to try to understand how the brain controls behaviors that might compel these kinds of cognitive interpretations. I want to just address the question here because it comes into the discussion we've been having about categories and stuff. So do we need emotion and other folk psychological concepts? I don't think we need them for most behaviors. Reflexes, certainly not for reflexes and innate behaviors. Now, innate behaviors or fixed action patterns, or what basic emotion theorists call basic emotions, are not emotions. As Lisa said, these are cognitive interpretations, or she puts it, constructions of situations. We don't need them for habits, and possibly we don't need folk psychology and emotion for instrumental goal-directed behaviors. I think they're needed, at least in the present state of where we are, for understanding subjective experiences and behaviors that are controlled consciously. So we live our mental lives and social lives in a folk psychological sphere. If we come up with a new language for emotion, it will need to somehow reflect the experiences we know of as fear, sadness, anger, guilt, love, and jealousy, and compassion. I mean, science brings new language into the culture, and that's fine. But it's going to have to be a language that reflects those experiences in order for people to use them to convey their experiences and to understand their experiences themselves. So here's a hypothesis. The brain mechanisms underlying emotional states of consciousness are not different from those underlying any other states of awareness. What's different about emotional and non-emotional experiences and different kinds of emotional experiences are the inputs processed by a cortical higher order consciousness network. That's the thing I pointed to up there. So here's a kind of depiction of what I have in mind here. On the left side of, and think of this as kind of a clock, on the left side of a clock, we have lower order elements that can contribute to emotion states, and that usually do, like sensory processing, semantic memory, episodic memory, self-schema. And on the right side, we have what we can call other lower order ingredients that can contribute to emotional states, such as the activity of a survival circuit. Now, I just want to point out that there are lots of, and this goes back to something Lisa mentioned, that, you know, the multiple ways that we can experience fear. You can be afraid not just because a defensive survival circuit has been activated, but because you're on a mountaintop and you have no food. So your energy management system is generating signals from your body to your brain that are being interpreted in a way, maybe I'm going to die. Same thing if you're dehydrating, maybe I'm going to die, or having thermoregulatory problems. Maybe I'll die from hypothermia. Reproductive, we could probably make a story there, but I'll skip that. So the survival circuit provides information into the blue spot here too. Arousal circuits do as well, body feedback, and emotion schema. And what is being integrated here in a very kind of complex higher order way is information that's being monitored, appraised, attended to, that we're having metacognitions about, introspecting about, and able to give verbal reports about. I think two important elements in all of this, which are kind of funnels through which the left side and the right side are passed in a way to get to these cognitive interpretations, are self-schema and emotion schema. And the survival circuit probably is often there as well, especially in a case of, you know, fear of a physical threat, like a snake at your feet. So here's the basic kind of model that I work with here. So we have a physical or social threat in the environment. It's processed by a sensory system. The sensory system is activating a threat detector that produces behavioral responses, behavioral and physiological responses. These are feeding back to the threat detector, but also entering into this mental model here through feedback to other circuits. Now, the sensory information goes into the mental model, pass-through schema as well, but so do the sensory combined with memory, or like we might traditionally call perception, also is going to pass through here, and especially through schema when we're talking about memory. So the schema are kind of a preconscious element of a preconscious mental model. And my idea is that the output of the mental model is something like a mentalese, a kind of abstract language of thought, something along the lines Jerry Fodor talked about long ago, but I'm changing it a bit here. And it's about generating a narration into emotional experience, and a separate narration. Well, the narration is one, but it's divided into two paths. One goes into the emotional experience and the other into self-report. And this can account for why, you know, we can't always put into words exactly what's on our minds consciously, and why self-report is somewhat slightly off, or can be very off from emotional experience, but often is very on. And the idea again is that the self-report and the narration involve additional processing, post-narrative processing, through the pathways that generate the experience and the self-report. And there's also noise in each pathway, which will also introduce variation in what we experience. So my idea about what an emotion is a mental model-based, narrative-driven, culturally shaped, subjective experience in a biologically or psychologically significant situation. So, sorry, this already in there. So what this leads me to is danger, not fear, is universal. All humans must deal with danger, and all cultures have words for danger and words for experiences that occur when we're in danger. But emotional experience varies across cultures. Because we can translate words like fear across cultures, we assume that people worldwide have the same basic experiences named by the word. Cultural differences in experiences are based on differences in the schema involved. Schema are personal, so that different people within a culture have different schema and different experiences. My experiences are not your experiences. We've all been exposed to different dangers and different kinds of situations. So we can't all have the same exact kind of conception and therefore experience of fear. Other animals lacking our specific kind of cognition and brain can't have our kind of schema and our kind of fear, but they may have their own emotions made possible by their brains. And in a piece in Current Biology in 2021, I've written about what emotions might be like in other animals. And I expanded on this in the Phil Trans article that was edited by Ben and Paul. As soon as there was life, there was danger, the deep history of our survival behaviors, and the shallower history of consciousness. So thank you very much.
All right, thank you very much. That was a wonderful talk. At the risk of putting it just a little bit over, I'm going to ask you one question that we got from the audience.
Okay.
And then Paul's going to start. If emotions are cognitive interpretations, whereas, for example, the threat and defense system serves the real survival function, what role or function would emotions have? And would this cognitive interpretation or feeling be able to influence those survival systems?
So first of all, yes, the emotions do have a function, in my opinion. They open up a whole another world of decision making and interpretation, and they allow. I believe in free will, so I think that we can consciously use our emotions to control our behaviors. We don't always do it very well because they're being competed with by these subcortical systems. They're on a different operating plan and so forth. And so they're going to be constantly challenging what we're trying to do consciously. But I do think that they are, that we both are consciously making decisions and also controlling behavior.
All right, excellent. And we're going to get a chance to ask more questions to Dr. LeDoux after a few more talks when we do our final open session. But to keep things on schedule, I'm going to switch over to Dr. Paul Cisek at the University of Montreal. Paul, are you ready?
Yep, I'm ready.
Okay, great. Paul is a professor in the Department of Neuroscience at the Université de Montréal. His lab studies how the brain makes decisions about actions using computational modeling and neurophysiological recordings in the cerebral cortex and the basal ganglia. Go for it.
Thanks, Ben. So I'm going to essentially echo some of the points that have been made by Lisa and Louise, and I think also the points that Patricia Churchland has made for many years, and that is really how to subdivide the brain into real biological categories. And I'll start with a common assumption, particularly common in cognitive neuroscience and systems neuroscience, that the brain is best described as an information processing system, whose principal function is to represent and transform information. And this applies both to sort of classical Newell and Simon type symbolic architectures, as well as to more modern neural network type models. And the basic reason why this is such a good way to think about the brain in terms of information processing functions is because if you can measure the input and the output signals of a system, then you can infer the computation that happens within that system, whether it be symbolic or a neural network type transformation. And if that computation is very complicated, then you could break it down into subsystems that process information. And then the intermediate representations between the subsystems form very good predictions about what you should be able to find in the brain through various recording methods. And so this provides a sort of a way to break down large problems into smaller ones that have predictive power. And this, of course, allows us to decompose a big problem like behavior into smaller problems, each of which can be seen as information processing functions, giving us a kind of a taxonomy for studying the brain. So you can then specialize in various systems. This taxonomy defines the questions we ask: how are memories stored and retrieved, how are decisions made, and then how to interpret particular brain regions in terms of these kinds of functions. Now, as many, of course, have already said, these concepts don't really fit the brain so well. In some cases, they're successful, but more generally, we see things that are not what one would expect. So, for example, things that should be unified appear to be distributed. We haven't found yet a representation of the external world inside the brain that's unified in some way. We have dorsal and ventral visual streams. We have multiple maps of space leading to what's known as the binding problem. How do you put this all together into knowledge? Conversely, things that should be distinct appear to be mixed. So, for example, you'd expect sensory, motor, and cognitive variables to be encoded distinctly, but in some regions, like here, this area LIP, you have cells that under one set of studies appear to be encoding attended stimuli, sort of the input to cognition, and under other series of studies appear to encode the intentions, or sort of the decisions made by a cognitive systems, the output. And finally, things that should be serial in principle appear to be parallel. So you'd think you make decisions before you plan actions, but in fact, what you see is that decisions and actions appear to emerge in parallel across extremely highly distributed regions of neocortex, basal ganglia, superior colliculus, etc. So one possibility is that we're just not looking at the data correctly. We need to measure it differently. But one possibility maybe is just that the traditional model is just wrong. It's just these categories aren't really the right way to subdivide the brain. And I think one reason to consider this a possibility is when you think about what the origins of these concepts are. They're actually very old. In particular, the distinction between perception, cognition, and action, I think, derives directly from dualism, the old philosophical idea that the mind is non-physical. This idea forced early philosophers to conceive of interfaces between the physical world and the non-physical mind: perception, which presents the world to the mind through the lens of attention, and action, which plays out the mind's intentions onto the world. Now, of course, this idea of the non-physical mind has been rejected, but the architecture that it led us into has nevertheless remained, and in part, I think, because these are just the terms we use to discuss behavior. And of course, I'm by no means the first to say this. And in fact, Paul and Patricia Churchland have been saying this for many years, that we have these categories, but how do we know that these are the right ones? Lisa Feldman Barrett has said this as well. Many others, Michael Anderson, Russell Poldrack, Yuri Buzaki, as well. Luiz Pessoa has just said this early this morning. The problem, of course, though, is that these are not the terms. Well, then what is the alternative? We need some kind of taxonomy, I think, because it's a big problem. So what is the alternative? How else to think about what the brain is doing? And how else to subdivide the big problem of behavior into more manageable problems that we can actually address empirically? And here I would just like to point out that it's true that we may not know what the really correct categories are, but we do know where they come from. I think we can all agree they come from evolution. And evolution really rests on two principles, as Ben pointed out in the introduction. It's not just natural selection that's important to consider, but also descent with modification. And this allows us to try to discover the taxonomy by following this process and trying to reconstruct its history. So very quickly, I just want to dispel some myths for those who are not perhaps so familiar with evolution. There's an idea out there, particularly in theoretical fields, that evolution is nature's solution to problems posed by the world. So it's like an engineer out there solving problems, but doing it with a particular method. This isn't really a good way to think about it. In part because evolution doesn't actually identify problems at all. It just modifies a functioning ancestor and then just favors those modifications, those variations, which happen to accomplish something and do a little bit better, solve essentially a problem. And importantly, for a variation of an ancestor to even enter into the game of natural selection, it must first be possible as a viable extension of a functioning ancestor. And importantly, and here's what's important, is that most variations are not possible. They're highly constrained by the ancestral system, in part because the genome is not a blueprint for the body or a connectome of the brain where you can just rearrange the weights like you might do in a genetic algorithm. It's a recipe for constructing the body through a series of stages. And like any recipe, each stage depends on the previous one. So you can't just modify some early stage without violating the assumptions on which later stages depend. So evolution can really only do a few things to a functioning ancestor. It can elongate certain developmental lines. It can differentiate and then specialize different things. It can back up and restart, but it can't just arbitrarily reorganize. So in other words, the mechanisms that we see in a descendant are highly constrained by the history of ancestors. Now, I would say this is actually good news, because that means first that we have a hope of reconstructing the history, but also it actually gives us a potential taxonomy of what the functional subdivisions are. So that's what I'm going to do. I'm going to sort of follow in the footsteps of evolution through a process I've called phylogenetic refinement, but it's really nothing more than applying the comparative method that's used throughout biology, applying it to theoretical neuroscience. And this lets us avoid sort of predefining putative systems like cognition, attention, decision-making, et cetera, and then seeing everything through that lens, but rather consider how the mechanisms were differentiated and elaborated over evolutionary time. And when I mean mechanisms, both the functional and the anatomical and the biochemical aspects of it. And I think this is useful because I think we can all agree that all distinctions between subsystems have some past, some history in which they were not distinct at all, because all these subsystems emerge from within a functioning system. And this is a very powerful constraint for theories, I think, and it helps. This is good because it helps us narrow down the space of theories worth pursuing. So to do this, of course, we need to follow a chronological sequence because each step defines the context for the next. Now I'm going to do that, but first I want to just sort of foreshadow two controversial claims that this, I think, leads to. And this is just my opinion. Well, no, many people have said this before. Okay, the first claim is that the brain is actually not best described as an information processing system, that it's better described as a control system that's regulating the animal's state. Really very much what Lisa Feldman Barrett already described, kind of a metabolic control, aspect of metabolic control. In other words, it's not the task of behavior is not to produce the right output for some input stimulus, but rather to control the input via output through the environment. Effectively, perceptual control theory of William Powers and others. And this isn't actually controversial at all, right? I mean, I don't think anyone would really disagree with this. Of course, the whole point of having the brain is to do something in the world to affect your state. I mean, right? I mean, so I don't think this is actually a controversial claim. We can discuss this if somebody wants to, if somebody would like to. I would point out that a control system is really a more specific thing than an information processing system. And that's good, again, because it has particularities that are important. And again, it narrows down the space of potential theories that one might want to explore. But anyway, this claim I don't think is really that controversial. It's the second claim that's actually more controversial. And that's simply saying that I think accepting claim one changes everything. In particular, the functional taxonomy, the kinds of representations and mechanisms we may posit, and the questions we ask. And I think this is the sort of thing I'd like to try to convince you of. All right, so to do so, I'm going to take this very brisk walk through evolutionary history. I'm just going to follow along the phylogenetic tree, along the lineage that leads to humans. I'm not going to say too much about other animals, except insofar as to constrain these stages. And it's going to be much too fast. So anyone who actually wants any of the details, these are some of the papers where this was published. I'm going to start with the origins of life, 3.5 billion years ago or so, which began with autocatalytic sets, sort of self-sustaining chemical systems that effectively produced replication and a primitive type of metabolism. Now, once these became enclosed in a membrane, you can now distinguish two types of metabolic control. Metabolic control that's entirely within the animal, essentially exploiting chemical processes to control, let's say, some nutrient state, and that we would call physiology. But sometimes there are some nutrients that you can't produce internally. You have to absorb them from the environment, and sometimes you will find yourself in a place where you don't have enough of that stuff. Well, one thing you can always do is just move, even randomly, assuming under the assumption that there's more food elsewhere or more nutrient elsewhere, that will bring you to a better state. And this is essentially also a kind of metabolic control. It's just that one that briefly extends to the environment. Then we would call that behavior. And many people have said that behavior is effectively this kind of type of metabolic control. You can think essentially of a behavioral control system as taking a nutrient state and a required nutrient state. Any deviation is some kind of a drive or an impetus for action, which motivates actions that then have certain outcomes that eliminate that impetus or reduce the drive. And the task of behavior, therefore, is fundamentally not to represent or build knowledge about the environment, but rather to complement the dynamics of the environment such that the organism environment system flows towards desirable states. I just want to point out that, again, by no means this is a new idea. Dewey said this over 120 years ago. John Dewey said, what we have is a circuit. The motor response determines the stimulus just as truly as sensory stimulus determines movement. Again, I don't think that's really controversial. The nervous system of early multicellular animals consisted of a nerve net distributed across the cup-shaped body with two specializations: an apical nervous system, rich in chemosensitive receptors, et cetera, that sort of did a high-level state control, and a blastoporal nervous system around the blastoporal involving synaptic transmission that controlled sensory motor activity, motor activity, sorry. They engage in this in a kind of a hierarchy with the apical nervous system doing sort of high-level modulation of the activity of the lower level ones to accomplish these goals. So we can now essentially take this basic circuit of nutrient balance and break it down into something a little bit more sophisticated, where if you're hungry and there's food present, then you want to engage in certain actions which sort of ingest that food and keep you in that region. That has the outcome of improving your nutrient state and also depleting the food. If you then find yourself, you're still hungry and you don't have food, well, then you engage in other kinds of activity like moving more long-range movements, which then hopefully bring you to a state of higher food source. And then the idea is that each of these actions is sort of a specialization that's governed by the apical nervous system secreting various neurotransmitters to control the particular patterns in the blastoporal. Now, notice what makes this kind of circuit adaptive is that the total sign around every loop, each one of these loops, the total sign is always negative, which means it's a attractor. It's a stable system that's going to arrive at a desired state. And notice what happens, though, if we ignore the environment's contribution, those gray lines. If we ignore the environment's contribution, we don't see that anymore. We don't see the adaptive nature of the system. And Dewey said this again 120 years ago. The reflex arc theory, or input to output, gives us a disjointed part of a process as if it were the whole, where in fact, what we have is a circuit. Okay, anyway, in our lineage, a number of interesting things happened. The body elongated, the apical and blastoporal nervous systems merged at one end of the body, creating the head. The body inverted dorsoventrally and folded into a tube, creating the basic architecture of the nervous system, where the the apical nervous system and this merging area became what we now call the hypothalamus, and the rest became the rest of the nervous system, primarily the spinal cord. And again, with this kind of high-level projections to the low-level controlling things like circadian rhythms, nutrient balance, etc., and locomotion being controlled by this other segment. These animals also possessed an escape circuit, very simple rudimentary escape circuit, where if a photosensitive patch projecting to a structure called the tectum means would essentially implement that if a shadow falls in the patch, then you quickly swim away. And that, of course, is also a negative feedback control system that eliminates this condition of being threatened. Now, this eye patch split into two, but migrated to the sides of the head, creating a kind of an oriented escape response, which was very, very important in these turbulent times of the Cambrian. Expanded into a retinotectal map, controlling the escape behavior more carefully, and then a specialization of this system with contralateral and contralateral projections back out to the actions, the spinal cord, that is effectively implemented approach behavior. So here I'm sort of building up the taxonomy. You notice I'm building sort of the functional taxonomy in terms of the history of these systems. This architecture can be seen in lamprey, zebrafish, as well as in mammals, where the tectum is called the superior ciliates. One major innovation in vertebrates was an expansion of part of the hypothalamus into the telencephalon, consisting of two segments, an external segment called the pallium, receiving sensory information about the state of the animal, On the subpallium, projecting downstream to control things like approach and avoidance through behavioral selection. And also this part now received a dopaminergic signal to reinforce particular state action pairings, effectively implementing reinforcement learning. And yes, this part of the circuit eventually becomes the basal ganglia. The pallium also elaborated a ventrolateral segment specialized for the sort of local interactions with the world, as well as the medial segment involved in more exploratory actions, navigations, etc. And this will become the hippocampus ultimately. If we continue, jaw vertebrates introduced the cerebellum for predictive, conferring predictive control and better sensory motor functions. I'm going to skip through some of the innovations of the bony fish and just go right into tetrapods, which upon getting out on land encountered a more complex world, differentiating the pallium even more into a ventral segment, a lateral segment, and a dorsal segment that sort of constructed different aspects of the state, the external key stimuli, interoceptive signals, as well as affordances, or what Gibson called affordances, the opportunities for action in the world. This part here will become much of the neocortex. So in mammals, this all expanded quite dramatically. We have the hippocampus here in the medial pallium. We have the neocortex in the dorsal pallium. And the neocortex now increased its direct control downstream. So now, rather than just detecting affordances, it controlled what Graziano and others have called action maps, or different types of behaviors guided, let's say, by visual information related to things like foraging. So searching for food, ingesting food, and things in between, like burrowing for food, breaking open a nut, or etc., called handling behaviors, as well as different kinds of defensive behaviors like freezing or escape. Likewise, this other more ventral system elaborated as well to collect more information about external key stimuli and internal states, as well to evaluate the animal state, producing a variety of types of selection signals. One, to select a particular type of activity, hypothetically through the basal ganglia, as well as activity within a particular system, selecting between specific movements. In primates, this expanded even more, dramatically curling now the brain around the insula into this familiar C shape that can still be interpreted in these general terms, except that now we have a larger number of different specializations of different action types, particularly within the handling system here. All the different things primates can do. Likewise, an elongation of this sort of key stimulus processing system to do something much more like object classification, object recognition, still in the service of valuation and action selection, etc. So in humans, of course, this is a relatively simple primate-like brain. But what about humans? You might say, well, how can you take this kind of sensory motor architecture towards humans? What about language in particular? So language is often thought of as some kind of an information transmission system. But I think, and many people have suggested, that it can also be seen as a type of control. So I've described physiology as sort of control within the body, and behavior as control through the environment. Well, we can also exert control through other creatures. So imagine if you're this helpless baby and you can't obtain something by yourself. It just so happens that in the niche of helpless human babies, there's something called a parent. And the parent will happily do whatever it is that the baby needs. In fact, the baby can, simply by uttering something, can get that parent to control its state in a desirable way. And importantly, the parent is very sophisticated and very complex. And so you can get very complex effects, but they're also very easily controllable. And I can tell you, because I have a 9-year-old son, you can just utter a very simple cue to get that parent to do whatever complex thing you need. And the parent, furthermore, will train you which cues produce which effects over time. And of course, this is far from the richness of human language, but it's a sort of fundamental aspect of communication, that it's not so much fundamentally about transmitting information from one brain to another, but rather it's about persuading others to do things that you need. This talk is an example. I'm trying to persuade you to think about a different sort of taxonomy for thinking about behavior, not necessarily this, not this breakdown. Although notice that some things do still exist. Reinforcement learning is fundamental here. Many familiar concepts are still important within this viewpoint, perhaps in a different context. Importantly, though, this taxonomy respects evolutionary history, and it's compatible with this constraint that the distinctions emerge from within. And that's not easy for many theories. That's not true for the classical taxonomy. And actually, it happens to map to brain regions, I think, better, in part because it's constructed always based on data about brain regions and their evolution over time. And it's a different model of brain function. It's not coding and decoding representations, but control of interactions. Very much related to perceptual control theory. But I would say more than being simply reactive, I agree with Lisa and others who have suggested that you need to predict. You need to predict things and control them before they happen. So what she and others call allostasis. It's also very similar to some of the things Carl Friston has said influentially, that this reducing of impetus is like reducing the free energy. So it's not serial information processing, but nested control loops. You'll have correlations with the external world simply through dynamical coupling, but most the representations, though not all, representations shouldn't be expected to be descriptive, essentially some kind of a decodable mapping of the external world, but rather pragmatic. Things like key stimuli, affordances that are very much tied into the an animal's context and not really decodable in any classical sense. And then the basic message then is that the evolution of behavior is just a continuous extension of this kind of control. And this leads to different scientific questions. It's not how does the brain encode something, not how meaning is attached to symbols, and not even what is the optimal solution, but rather questions that mostly focus on transitions. Like, for example, how did the brain and behavior change upon the water to land transition? Something Malcolm McIver and Barbara Finlay have explored very nicely. How did prefrontal cortex, where did it come from? How did it, and one proposal, it extended premotor functions in some way. This is something Ben Hayden has talked about, as well as my colleague Giovanni Pazzullo in Rome, who's also asked the question, well, how do symbols, it's not how meaning is attached to symbols, but how do symbols emerge within grounded interactions? How do they detach from those interactions? These kinds of questions. And then, a question that, of course, everyone would ask is, okay, what about human thinking? These kinds of things like how did, system 2, cognition, et cetera, emerge within this interactive brain? And I don't know, maybe you have the answer to this, but I would just claim that maybe this cognition and system 2 is not so much a system at all, but rather a strategy for using these ancient circuits. So with that, I'll thank you and take questions.
All right, thank you very much. I'm going to start. We have 2 minutes, I believe. So I'm going to ask you one question, and then we're going to move to the final speaker. Here's your question. What is the problem with umbrella concepts such as memory or attention if we later use enough subfunctions to find a one-to-one relationship? For example, we use perception and then we find the grasping network. I don't really get why we cannot maintain a classical model and study neuroscience in the way that you say.
Well, I think in some cases we will arrive at those categories. Like we could say we can find the grasping system in the classical sense as well. But I think in other cases we don't, or we struggle for decades with these false dichotomies. And one example that I've often raised is this attention-intention debate regarding parietal cortex, which has been the subject of 3 generations of neuroscientists now, trying to decide which of these functions the parietal cortex is doing. And I think that's because it's a false dichotomy. And neither of those terms actually describes the parietal cortex well. But I think an embodied perspective leads to that better. And of course, I'm not the first to say that, but I think there are many cases and other cases in the question of, does basal ganglia contribute to decision making? And I would say, well, if you think decision making is one thing which has a system associated with it, then there is no good answer to that. But if you think about the kinds of decision systems that emerged, one of them is the basal ganglia, and it deals with certain kinds of decisions, but not others. And so I think certain dilemmas and certain issues dissolve. The binding problem, I think, is another one, which exists in the classical, but it just dissolves in this sort of alternative taxonomy. And I could give other examples, but I don't want to take up too much time. Barb had a question. I'd love to hear what Barb has.
Okay, am I on? Yeah, I like the sort of derivation kind of argument in general, but I am a little concerned that you go so general and abstract that you will have the danger of quite often just missing what has actually happened. For example, you could talk about what happened to the vertebrate pallium when animals go on to land as extension of the properties of the pallium, but as Malcolm and I discussed, you've got a very specific problem. You've got to control movable eyes and head and know where you are, do those computations. You have to control limbs and all the degrees of freedom that those insert in some particular mechanism. And what we discussed and looking at that if you try to get to not the very specific exact behavior, but what is this new set of computational issues, then that moves you a lot farther along in understanding what the fundamental organization of the structure might be than just we're going to extend the control features of the pallium. I'm afraid that kind of language just gets us right back to, and we get better and better, which is it's sort of the amelioration version of your whole thesis there, and it might not really be, it might be just one level too far up to capture what kind of changes actually do occur.
You mean I focus too much on gradualism as opposed to something?
Well, gradualism and just that the fact that you would always find it acceptable to say, okay, we're going to expand the pallium, and it's going to, by that alone, it will make better, more informed control over behavior. But if the real thing that has happened is that you've developed a lot of circuitry for limbs that then can be exploited, you've got a very different account of
well, I mean, I certainly think that the circuitry for limb control is one major innovation that has probably very little to do with the pallium per se. Well, I mean, in terms of the spinal circuitry, there's a whole.
No, I mean, the control. But that's one of the major.
And that's a major innovation. I didn't mention that, but that's of course very important. The changes that happen in the spinal cord are actually extremely important for the actual getting out on land and maintaining body integrity and things like that. But if you look at what seems to have happened to the pallium, the medial pallium seems to have expanded a lot more at first. Was one of the first regions to really expand, and still is relatively large in amphibians. And if that is the sort of the navigation center, that makes a lot of sense in terms of, one of the fundamental things that amphibians had to get right, or they wouldn't have done very well, is find their way back to water, et cetera. And so that is a very major aspect. Other parts of the pallium probably didn't expand for a long time, especially since these animals relied, I think, much more heavily on the optic tectum, and that probably expanded a lot more than these other.
No, that's exactly what we argued could not happen. That the optic tectum is very much sort of fish body centric, and it never incorporates in any animal I'm aware of things like limb control and so forth. It stays in the approach thing. So that's why it needs a different, and what the—
So you're suggesting that getting and controlling that body required pallial innovation.
Yeah, it required,
well,
it required some different structure, because the midbrain one appeared to stay very conserved, and you only do a very minor amount of correcting for eye position when you're talking about approach avoidance of the whole body, and all the other machinery is very much distributed around the pallium, the cerebellum, other parts of the midbrain. So are coral reef fish getting back to its hole different from an amphibian getting back to the water?
Well, they both have to. They both deal with that problem.
Yeah.
Sure. I think
I
think the complexity, though, of the one of the things that you get on land, though, of course, as you've emphasized, is long range vision, and so now landmarks, using landmarks for navigation becomes really powerful, whereas previously you really had to rely more on autogradients or immediate visual cues, saying this is the hiding spot. Here you can actually use treetops and mountain tops to guide yourself, and so that's a big difference in being out on land.
Yeah. Okay. Yeah, well,
All right,
I'm making an argument for more specifics.
And there'll be plenty of time to have these discussions, and I'm actually looking forward to those discussions. But just for all the people tuning in, we're gonna move on to our final speaker in our series, and then we're gonna have our discussion. Patricia Churchland is the Emeritus Professor of Philosophy at the University of California, San Diego, Adjunct Professor at the Salk Institute. She has been awarded the MacArthur Prize, the Rossi Prize for Neuroscience, and the Prose Prize for Science. I'll also say that she's extremely influential on my own thinking. When I was an undergraduate, I only took one neuroscience class. It was a philosophy of mind class. And I didn't do so well in the class because you had to write essays that kind of presented both sides of an argument. And as soon as I learned about eliminative materialism, I really was no longer able to take both sides of the argument. And I basically have been a convert ever since, and I still believe in that philosophy. I'm looking forward to seeing what we're going to hear about today. So, Dr. Churchland, are you ready?
I am, except that I don't seem to have a way. I hit screen share and it's not really, okay, so that's what I've got. I just click on that.
No, we can see your screen. You just need to go to your PowerPoint or whatever and just.
Okay, now can you see my PowerPoint?
Yep.
Yep.
Oh, jolly good. Okay, then I think.
You can just start your slideshow and we'll be seeing it, I think.
Yeah, okay. Yeah, there we go.
Perfect.
Okay, well, first of all, this has really been a wonderful morning or afternoon, I guess, for some of you. And it is very exciting to see how much we can learn from understanding the evolution of structures in the nervous system. I don't seem to be able to see my screen.
Yeah, we lost it for a second.
Okay, can you see it now?
I can't.
No.
You can't. Well, okay.
It stopped sharing somehow.
I think
Yeah, maybe I better try sharing again.
Yeah.
Okay, isn't that funny? All right, we'll try again. So I hit share.
Yep.
Yes.
Okay, and then if I go to a regular slideshow, that seems to be when it disappears.
No, it was there.
No, I think he was fine for a second. Maybe something.
Oh, good. Okay.
Yeah, he did
Well,
look
I was
fine.
in the middle of my thank yous, and it really is an enormous pleasure to be here. From all of you at various times, I have learned so much. Today, I'm going to sort of stick my neck out a bit, and part of what I have to say about the evolutionary origins of mammalian sociality will be hypothetical, but to the extent that I can draw on existing data, I'll try to do that. I'm going to start actually by saying a little bit about, Barbara, you have your hand up. Are you okay? Okay.
Sorry, Pat, could you hit the slideshow, play slideshow button again? Because I think.
Oh, yeah, well, that's what I was wondering about. Okay.
I don't think that she selected the screen that she wants to share. I think we're just seeing the one.
Try that.
Accident, sort of.
I think it's
How
good.
is that?
I think it's good.
Because I'm not seeing anything.
No, it works. We see it.
Yeah, I see it now.
Can you see that, Barbara?
I can see it, but it's not. It's
Not very good.
small. I don't know.
It's small.
No, I think it's the whole screen, right? I mean,
I'll go. I'll try.
I think this.
Try advancing your
It
slide.
goes to the whole screen.
That's
I'm
better.
sorry.
That's better. Yeah, got
Do
it.
you want me to start again?
No, fine.
It's good.
Okay.
Mhm.
Okay. So I'm going to start with a sort of brief overview of recent work in moral theory from the perspective of philosophy, just to sort of orient us to how philosophers in the recent past have thought about these things. But then I'm going to go to the sort of more ancient philosophers such as Aristotle and Confucius, who I think basically had a more reasonable take on things. So moral theory from about the 18th to the 20th century really had a fascination with rules. Now, prior to that, of course, there were theological philosophers who had views about the nature of morality, such as Aquinas. But these were all very much attached to what God wanted us to do, what God instructed us to do, or very special books that would tell us what the rules are. But interestingly, in the 18th century, partly as a result of Hume, David Hume, the Scottish philosopher, who was clearly unimpressed by the idea that God had much of a role to play in social behavior, philosophers started to try to develop a system of rules that would guide morality, that would be exceptionless in the way that theological rules were, but that didn't involve an appeal to God. And Immanuel Kant is sometimes assumed to be the sort of bedrock of this. And he really, although he did think that God was important and was a theological believer, he thought that the foundational rules came out of reason without any kind of appeal to emotion or feeling. I'm using those words in their full psychological sense, or feeling or anything of that kind. And he thought that the rules could be tested for whether or not they were moral rules by asking whether the rule was rationally universalizable. That is, could you say that this was a rule that everybody should follow with no exceptions? And of course, people with any kind of wit at all began to dream up exceptions to rules such as always tell the truth, or always be compassionate, or always be courageous. And so he was really not, I think, taken nearly as seriously at the time as he was by 20th century philosophers, especially at Harvard. Now, the other thing that was quite interesting, and this was really developed by some of the Harvard philosophers, was that if you took any pleasure or joy or satisfaction in doing the right thing, then it didn't really count as properly a moral decision. So it was very tough. Now, there is still, I think, amongst philosophers, determination that rules of some kind are absolutely the bedrock of morality. And as we'll see, this is really at odds with Aristotle, Confucius, and later David Hume. But the point of view that rules are critical also came through in the 18th and 19th century, especially by the English philosophers, for example, Sidgwick and Jeremy Bentham, who were utilitarians, who thought that the right thing to do was to follow the rule of utility that says maximize aggregate utility. And they thought this was true of our actions individually, as well as the actions of a government. This too, of course, was very easily shown to have all kinds of weaknesses. You might, for example, maximize aggregate utility at the expense of a small minority, so that you might have a rule that said it's okay to enslave all red-haired people because most people aren't red-haired, and so you can maximize aggregate utility in that way. And as you will, most of you will know, because most of you probably took a philosophy course at some point as undergraduates, one of the things that you can do is to try to take in a very fair and serious way utilitarianism and show that as an exceptionless rule, it really won't function. Now, of course, there's something right, both about Kant saying that if a rule holds for me, it really should hold for everyone. And there's also something right about utilitarianism that stresses that the consequences matter, whereas Kant thought the consequences didn't matter, that really all that was important was rational universalizability. But we all realize that very often the consequences matter. But the trick that failed was to try to get an exceptionless rule. Now, Aristotle, and as I said, Confucius also, Aristotle took a really rather different view. He didn't know anything, of course, about Kant and the utilitarians. He was a very practical man. His father was a physician, and as a child, he helped his father. He was really unlike Plato, who was constantly about the abstract realm of reason and purity and perfection. Aristotle was really about understanding the world around him. And whereas Plato thought that the abstract rules were in some abstract realm of reason, Aristotle took a very different view. He thought of ethics in a practical way. He distinguished between ethics as a practical matter and physics as a theoretical matter. And he thought that acquiring ethical dispositions or acquiring norms was a matter of acquiring a social skill. And that may be the idea of a skill, incidentally, may be one of the folk psychological terms that we don't want to jettison. He thought that by and large, as a first approximation, what we should do is avoid extremes. You don't want to be too humble, or you don't want to be vainglorious. You want to be in the middle. You don't want to be a coward. You also don't want to be rash, but you want to be appropriately courageous. And of course, what that exactly means is a difficult thing to explain, but it can be, he thought, a skill that you acquire over time. You interact with other people, you model yourself after people who are living well, surviving well, doing practical things, and are socially appropriate. And we see much the same thing in Confucius. He thought too that ethics involves social skills, that we do well when we are young to model ourselves after an exemplar of the community who understands how to get on in life, especially how to get on in social life. Now, Hume was really more like Aristotle and Confucius than like the 19th century philosophers. And in the 20th century, Hume was largely eclipsed. But what Hume thought was that morality is rooted in our social nature. Now, I want to go on from there to contemporary times. And famously, Ed Wilson said in 1975 that the evolution of human sociality is the fundamental conundrum of biology. Now, whether it really is the fundamental conundrum, I don't know, but it is a conundrum. Now, as many of you have remarked, it's an outcome of natural selection and descent with modification that all organisms have the value of themselves. They care, so to speak, they care about their own survival. If they don't, of course, then they don't survive and don't reproduce. And this has led many people to think, as Richard Dawkins did in The Selfish Gene, that we are born selfish and that we kind of have to have generosity and kindness and compassion and altruism beaten into us because it goes against our nature. And if that's true, then it is kind of hard to see how it's ever possible really to motivate anyone to be a moral agent. Now, it's interesting to me that Darwin had a very different view. In The Descent of Man, he says that our social instincts, that our sociality really depends on our social instincts, on habits and skills, and our capacity for problem solving. That is, when we encounter a very new ecological or social situation, we are able to have the flexibility to accommodate ourselves to that. And as I indicated, this is really part of the long ancient tradition, not part of the sort of strict rule story that we see in Aristotle, Confucius, David Hume, and also actually Adam Smith. Now, within our own time, over the last 40 or so years, anthropologists, both those studying animals in captivity and those studying them in the field, have studied social behavior and discovered that not only does sociality not have to be beaten into these creatures, but that they very naturally show it. And so we see consolation, reconciliation after a squabble. We see pro-social choice, orphan adoption. Chris Besh has seen 5 distinct instances of a male chimpanzee adopting a very young chimpanzee after its mother was killed or died. 5 instances, and it was no small feat for those males to be able to do that. Were they maybe genetically the fathers? They took urine samples and they were not. We also see instances of empathy, as in this particular picture. We see punishment of the miscreants, self-control, cooperation, and reasoning. And this tends to suggest that there is something that is similar between the sociality we see, for example, in apes and in the human ape. Now, sociality, of course, is not unique to mammals. We see social fish. And even in the case of these discus fish, the baby fish are feeding off the outsides of the parents. And of course, we know quite a lot about the sociality, the wonderful sociality of bees. So the question is there something similar between them and us? And if there are differences, what are the differences? And I'm going to emphasize the differences, not because I think we're the highest and coolest of animals or anything of that kind, but just because there seem to be differences. And the main difference is perhaps that in mammals that are social mammals, we see a degree of flexibility in behavior that shows they adapt well to changes in circumstance, changes in the social organization, changes in the environment. Now, one hypothesis that I tend to favor has to do with the link between endothermy and the rise of social behavior in mammals. And I'll try to make that case as we go along. There may be many things that I don't know about that evolutionary development, and I'd be more than happy to be corrected on that. So with proto-mammals who were warm-blooded, there was a tremendous ecological pressure, and that is gram for gram, they have to eat about 10 times as much as their cold-blooded cousins. And so one question is, after the great extinction about 65 million years ago, there was this widespread species differentiation in mammals, and somewhat later than mammals are in birds. And the question is, what's going on here? Now, I'm going to pretend that I'm Mother Nature, not because I think anthropomorphism is a good thing, but because in the interests of time, it suits me to tell the story that way. So here I am, Mother Nature, and I realize that these endotherms have to have so much more energy. And I'm wondering how to sort of help them out. And the basic idea is that their learning capacity should go way up. And that will allow them to do things that they couldn't otherwise do. Now, of course, what they can do that the cold-blooded animals can't do is forage at night and live in cold places. So that's a great advantage, but they do need to be big learners. And correlative with that, basically what we see is a downgrading of neonatal independence. So whereas this wonderful turtle can scrabble out of its egg, get down to the water and take off, mammalian infants have to have a parent nearby. So in neonatal rats, for example, they're born hairless, they're blind, they're utterly helpless. All they can do is scrabble around, find a little warm thing and suck on it. Now, in order to achieve great expansion of learning capacity, the suggestion was that we see the development in mammals, not in proto-mammals or early sauropsids, but in mammals, we see the development of cortex. And as we now know from the work of Harvey Carton, who was at UCSD for a long time, we also know that birds have something very analogous to cortex. It just doesn't look like that. We won't go into birds for now because it's a big topic. And of course, cortex does seem like this amazingly highly organized, highly structured thing. However, in reading Strider and Northcutt to 2020, I discovered that the brain of the hagfish, very ancient vertebrate, actually has laminar structures. And so we can see laminar structures here and diagrammatically here. So what they suggest, Strider and Northcutt, is that when you have hypertrophy of a structure, you tend to get lamination. And so it isn't quite as amazing in the hagfish as you might think, although certainly mammalian cortex, neocortex, does seem with its 6 layers of highly regular organization to be quite remarkable. Now back to the learning point. So as Mother Nature, I said that it was going to be very important for these endotherms to learn, and that they needed to be born very immature in order to learn, because you learn by making structure. And in this slide, we see that the structure of cortex changes enormously over just very short time, so that by the time we're about 2 years old, there is already vastly more structure than there was at birth. But lack of structure means that the organism has to be born very immature, and you can't just selectively make the cortex immature. You got to make the whole thing immature, which is why those poor little rats look so absolutely helpless. Now, one of the things that we do see in all mammals and all birds is the expansion of the domain where the brain manages well-being. So that there is not only the organism that takes care of itself, but the organism is motivated, if I may say, motivated or wired to take care of others. Specifically, the others are, of course, mothers and babies. And all vertebrates, so far as I know, have both oxytocin and vasopressin. And in mammals, these are put to new jobs. And basically what seems to be the case is that with birth, there is this huge rush of oxytocin in the mother, which both involves the capacity for lactation and parturition, but also attaches her to the baby. Now, I don't know whether I should probably not use the word attach, but I'm going to because I don't know what else to use at this point. And then oxytocin is also released in the brain of the baby. And over time, the baby becomes increasingly attached to the mother. And as Paul pointed out, babies are also very good at controlling the mother. They will whine or cry or yelp as necessary to get what they need. Now, I'm just going to say a little bit about oxytocin and vasopressin because I imagine it's basically quite well understood. But it is released in the hypothalamus. And the important projections for my story are to the nucleus accumbens, where there is release of endocannabinoids. So not only is the release of oxytocin sort of involved in a direct way in attachment and bonding, but it kind of makes you feel good. If I may say feel. And there are projections elsewhere. In the slight blown up diagram, what we see is that there are really two main sections from which oxytocin is released, but notice that it's also released into the posterior pituitary. It's also the case that oxytocin is found all over the brain. And all over the body, actually. It's in the ovaries, it's in the testes, it's in the gut, it's in the vagus nerve. And so it is not unique to this special function in the brain. Now, what does this all have to do with morality? Well, I didn't think it had much to do at first. And then one day, Larry Young came to the Salk and gave a talk, and it had a very nondescript title that, sounds like, could be interesting. So I went, and it changed the way I thought about morality. It allowed me to take the next sort of biological step from the story of social skills that we see in Aristotle and Confucius to seeing something very different. So as you know, there are many species of voles. Montane voles live in the mountainous regions. They make their nests in amongst them, down under rocks and so forth. And in montane voles, the male and the female meet, they mate, and then they go their separate ways. He's looking for more action. She's going to have the babies. And then Larry Young tells us about prairie voles. And this I thought was very surprising. I'm used to it now, but it still kind of surprises me. In the case of prairie voles who live on the open prairie in these kind of large communities of voles, standardly the male and the female meet, they mate, and then they're bonded forever. They stay together, they share the care of the pups. The male guards the pups when the mother goes off to feed. The male also may adopt orphan pups. And lots and lots of research, of course, has been done on this, showing that if the bonded males and the females are separated, then they both get very depressed. and you can measure their oxytocin levels. It's not good. They get back together again. They're happy. Well, sorry, I shouldn't say that. They show happiness behavior. They bounce around, and they appear to be satisfied that they are back together. Now, as you know, the main difference that was found early on had to do with the density of receptors for oxytocin in the prairie vole versus the montane vole. And here you're looking at the nucleus accumbens in the prairie vole, the ventral pallidum in the prairie vole. And what you see is that the density of receptors for oxytocin is very high relative to the montane vole. And for vasopressin, it's relatively high relative to the montane vole. Although notice that there is a density of receptors in the lateral septum of the montane vole, not the prairie vole. And there is a bit of a story there too. Now, I think many of us who were in the room at the time Larry gave his talk were pretty naive about the social behavior of many animals and were stunned to find something that is often described as a religious necessity that you see only in humans, namely long-term pair bonding, is actually seen in almost all birds and in about 5 to 10% of mammals. And here you see marmosets who are intensely social and who stay together. They too are bonded for life. And I know there's other Canadians here like Lisa. You'll be happy to know that beavers also are long-term pair bonders. And that usually does involve sharing parental obligations and so forth. So I began to think that in a way, this extension from me to me and baby, but also from me to mate, and maybe not to mate, but to kin, as in baboons or as in chimpanzees. Maybe something like that is what would have pleased Darwin when he thought that there were social instincts. In this slide, I know there isn't such a thing as a reward system, but I need to show this slide anyway. What we're seeing are the projections from the paraventricular nucleus of the hypothalamus. But again, what you want to notice is that the nucleus accumbens is affected, the ventral tegmental area is affected, supraoptic, olfactory, amygdala, and of course, the pituitary. But the other side of all this is also really, really interesting. And that is that we see that there are oxytocin receptors all over the place. And so that tells us that whatever is going on here, we really have only got sort of the tip of the iceberg. Behaviorally, we know that oxytocin and stress are kind of in opposition. That is, when stress levels go up, oxytocin goes down. When oxytocin levels go up, the stress response decreases. We know that pleasant social interactions and even physical things like running around because you're happy, not because you're running away from a threat, increase the level of oxytocin. Food sharing has been shown in monkeys and in chimpanzees to increase the levels of oxytocin. Now, Sue Carter has also done a tremendous amount of work that's really careful and really thoughtful, and I greatly admire her work. And in her 2018 paper, she wanted to contrast the differences between when there's just oxytocin release or when there's both oxytocin and vasopressin or when there's only vasopressin. And what I think is quite interesting is that when both are released, we see these very complicated kinds of social behavior, sexual behavior, parental behavior. Vasopressin has more to do with defensive postures, aggressive behavior, and so forth than oxytocin does. And probably in all of these things, we see both approach and avoidance and all of that complexity that we know involves social behavior. I'll just mention quickly, this was a nice work done by Alison Perkybile showing this was in mice. So the strategy was to have the experimental animals removed from the mother for a few hours a day, 3 to 4, kept warm, kept fed, and then put back in the nest. And the control animals, of course, were always with the mom. And what you see is that in the experimental animals, there is methylation of the DNA for the oxytocin receptor. So you see decreased gene expression for the oxytocin receptor, and hence there is decreased binding of oxytocin because there are fewer receptors. What does that mean behaviorally? What they found was that species typical social behavior in mice was very different. The females who were removed from the mom in this experimental manipulation showed they were not regular standard mothers. They didn't really care that much about the babies. They didn't do the standard social things that the typical mice did. And of course we know, although we don't know in the case of humans often what the causes are, but we all know, probably even in ourselves, but certainly in others, that sometimes there are people whose social responses are really rather odd. And this is sort of the case in point. Now, of course, it isn't only primates that are social. I've already mentioned beavers, but famously, it has worked, sociality has worked really, really well for wolves. And they all are also long-term pair bonders. And in this instance, what you see, of course, is that this pack has cornered a moose mother and the calf. This one will almost certainly go for the leg and lame it, while this one will rip the throat out, and then they'll take the baby. And you can see that that's obviously a way of getting a much, much bigger meal than trying to be a lone wolf. And we know from the data that lone wolves often do not do very well, but wolves within a pack are great survivors as long as humans keep out of the way of their poison.
Okay, so we're looking at 3 minutes left.
Okay, so the hypothesis is that mammalian and avian sociability involves oxytocin and the cannabinoids, and that norms emerge from learning and being rewarded and so forth. So experience shapes our sociality into habits and preferences using the reward system. But norms are not like the old-fashioned Kantian rules. And norms, we all know, are very complex because they can conflict with preferences, with other norms. They vary across individuals and cultures. They vary within oneself across time. They involve memories. They are affected by such things as sleeplessness, hunger, and so forth. So the basic thesis then is that sociality begets other caring, and other caring plus social learning begets social norms for right and wrong. These are the 3 Darwinian points, really, that problem solving in ecology begets norm changes. And I'll just finish by saying that morality is not a module, but then cognition is not a module either. And finally, despite the fact that Kantians think that we're not supposed to feel joy or pleasure in any sort of moral behavior, and here by moral, I just mean incurring a cost to yourself in order to help another. There are lots of things that humans do, both in very complex societies and in hunter-gatherer societies, that are largely unrelated to passing on their genes. And we even see this amongst wolves, but famously, of course, amongst dogs. So I think social behavior is tightly tied in our evolutionary past to what we call moral behavior, but that moral behavior probably is best understood, as Aristotle and Confucius did, as instinct plus learning of how to get on, how to be a human in a group. And that's it for me. Thank you.
All right, thank you very much. What we'd like to do now is do a brief question and answer period where basically anybody can ask questions. We prefer if you go through the Q&A element of the webinar, and we will bring those up. Let's just start with the panelists. Before I do that, I see 2 hands raised among the panelists, and if I understand these correctly, how do I get to these? Oh, here we go. Let's start with Dr. Joe LeDoux and take his question. If I understand correctly, he has his hand raised.
Thanks. Pat, could you just clarify where you stand on eliminative materialism?
Oh, yeah.
I mean, I know you said something earlier, but just kind of round it up, because you have a lot of that in the talk.
The basic idea that Paul and I had in the 80s went something like this, that just as in other sciences, the sort of folk physics and folk chemistry changed profoundly in its categorical and explanatory structure as science developed, so it's quite likely that as neuroscience develops, we're going to come to think about folk psychology and as not really applying very well to the brain. I mean, I still talk about earth, air, fire, and water, but it's not like I think those are the elements. And I'll probably, even after we have a deeper understanding of what brains are really doing, that we will still talk about memory and emotion and skill and so forth in a casual context. But the idea was that, and we thought this was an empirical hypothesis. The idea was really just that we should expect there to be changes in this ancient thing, this folk psychology, when we really get down to understanding the neurobiology of how it works. That was basically the idea. We thought it was a really very bland idea, actually, but it caused no end of trouble. You're muted, Joe. I can't hear you.
So it's no longer something that you really want to push.
Oh, sure. I mean, I still think that it's essentially correct, but we started talking about this in sort of the early 1980s, right. and we said our piece, we gave the arguments, and it still seems quite likely. And I think both Lisa and others have really made a case for saying, we've got to be a little bit leery about applying certain kinds of concepts to the nervous system, as though that surely is true. We can do it in a kind of tentative way. And I think like Graziano, I think, is a good case in point when he talks about attention.
So, I mean, for me, I'm happy to talk about all these things in non-psychological terms, in terms of behaviors. But, when we get to subjective experience, it seems we need folk psychology.
Well, we may.
At least now we need.
Yeah, certainly right now we do. I mean, I'm perfectly happy to be with Dewey here and be a pragmatist. I mean, you don't throw away your crutch if you can't walk. Right. And so I'm entirely happy to use provisionally concepts that I think might be improved and revised over time, or maybe they won't be.
Okay, thank you.
I don't know. Consciousness, of course, is a bit of an issue, and you know, I think I have some disagreements about whether language is necessary for conscious.
I don't think, probably not, but
well,
I mean, I probably don't. I think we probably don't disagree that much. But
oh, good. Okay, because I think they're probably conscious.
okay, well, thank you very much.
Yeah, thank you.
All right, let's take Paul Cisek, who also has his hand up, and then we're going to move on to some questions.
So my question is really actually very short. I was intrigued by your connection between the sociality and parental care and warm-bloodedness, and immediately I thought of there was a paper I remember that I found quite good by Barry Lovegrove, describing the evolution of endothermy. And in both mammals and birds, he describes it in terms of 3 stages, the first of which has to do with parental care, actually, and locomoting over a larger area. And only after that, in both mammals and birds, do you get actual warm-bloodedness. So I thought that was quite an interesting link, and perhaps having in the end to do the metabolic needs and the ability to provide that high metabolism for babies, right?
I should sort of step back from that, and because I don't think it's absolutely known that parental care came before endothermy. There is some reason to think that you know, endothermy probably evolved a number of times, and I don't really understand it. I mean, some people talk about it in terms of leaky.
Well, he's basing it very much on fossil evidence and what you can infer from that, which, you know, I don't know how you can infer things from.
Anyway, I mean, it looks to me like there is a link, because there is this link between endothermy and parental care and this new structure that we see in mammals and birds, namely a cortex.
Anyway, I'll step aside and somebody else wants to ask questions.
All right. I'm just checking all these different things.
Pat, can you stop sharing your screen so we can?
I will stop sharing my screen. Yes. There we go. How's that?
All right. We have another question from the audience. Here's a question. I suppose that with greater learning capacity comes more energy-hungry brains, which sounds like a vicious cycle for the already energy-hungry endothermic animals. Would you care to provide some insights into this puzzlement?
Yes, and actually, Strider and Northcutt are very good on this. And it's certainly true that once you've got a much bigger brain, you've got all that cortex, you got to feed the darn thing. And it's very energy intensive. So it's really important that the animal will be smart enough to do well. And of course, as you know, initially, the endotherms all lived underground and ate insects, but once the great extinction was over and animals came out, and then there was this great speciation, then you see all kinds of differences, and I'm sure Barbara can help us a lot on this, and Lisa probably too, in the growth of brains. You also see the growth of hair, and in birds, feathers, hair and fur, and nasal turbinates to help warm the air so that you don't have to use energy to do that, and so forth. So there were many, many little changes that were also really, really quite important. And I should know more, but I don't.
All right. And remember, this is a general discussion for everybody. So I'm going to just throw this question out because it's not addressed to anybody, but I have some guesses about who might have opinions about this. What is the connection of the control theoretic way of looking at cognition and behavior to the predictive brain / Markov blanket / free energy principle, Carl Friston, way of looking at it? Are they equivalent? If different, does one base the other? Is one more fundamental than the other? And what's the connection? Well, let's stop there.
I think the answer is yes. They're very closely related, and again, there's a long history of ideas that are very closely related. Ecological psychology is there, cybernetics is there, the work in situated robotics is there's a lot of ideas autopoiesis of Maturana and Varela. There are many ideas that really hit on the same key themes. Most, sometimes not knowing about each other, sometimes knowing about each other. But I think in the terms of certainly the kinds of stuff that I talk about, I think Lisa can say so. I mean, she has already shown that her ideas are also very much in line with a lot of the things people like Carl Friston are saying, and Andy Clark, many people, I think, and I'm not sure one is more fundamental than the other. I think we're all sort of poking at it from different directions with sort of on the same wavelength, and emphasizing different parts. So for example, I'm coming around to the importance of predictive processing, et cetera. I don't think it was there in the beginning, but it became very important. It becomes very important for any control system to do that. And I think, again, there may be disagreements. I mean, I think Carl, my opinion, I think Carl Friston's got it all right. I think he's emphasizing the symmetry perhaps more than I would, because he talks about a process of inference or modifying your beliefs to fit the world as being sort of symmetric to changing the world to fit what you'd like it to be. And I think maybe there's an insight there, I think, which is very interesting. But I think beliefs are relatively recent innovations on the stage, and I think predictive control is perhaps more fundamental. But that's, anyway, other people should chime in.
I think the ideas have been around for really quite a long time, and they're very general ideas, rather abstract. And I mean, this might sound weird coming from someone who is trained as a philosopher, but I'd really like some detailed explanations of some actual phenomena in the brain. I would like to see Carl Friston, for example, tell us something in detail that isn't just this large scale waving. Yeah, okay, the waves are really nice. We like all that. Now go for it. Give me something I didn't already know.
Well, I would say that some people have done that. I think that's probably better done when you get away from some of the elegance and generality of it. Only then can you really start getting at the nitty gritty. So I think that's maybe some of his work is trying to be too general and too elegant mathematically to be able to answer the question the way you want to hear it answered. I think we have to give up some of that to get at the nitty gritty of actual circuits in the brain.
Well, you might have to, but it seems to me the time has come. I mean, Friston has been shopping this now for 25 years. Can you give me something more than generalities?
We'd have to ask Friston. But I think maybe Lisa can say a little more.
Yeah, maybe. Okay.
Well, I think that it's probably not a good idea to, I mean, I think, Paul, you're correct that all the things you listed, ecological psychology, cybernetics, and so on and so forth, they all bear some similarities, and that's really true. But even within just the literature on predictive processing, there are real differences that are at the circuit level, at the computational level. Not all predictive processing is Carl Friston. I think that's really important to understand. No knock to him at all, but just that's one view. And there are many views, and some of them are actually pretty detailed. So I think they would, they would satisfy, they're talking about measuring individual neurons, talking to each other temporally. So I don't think that there's enough, for example, some predictive processing models, make a distinction between, they're talking about like there are regions of the brain or even particular neurons for prediction versus prediction error. Whereas there are other models where every neuron is doing some integration of both, types of information. there are real differences, and but I do think that there's more evidence now, maybe in the last 5 years or so, for specific hypotheses, not all of which are at this really abstract level. So for example, you know, the Friston approach really relies on the notion of homeostasis, which is reactive change, you know, like a reactive change to bring a system back to a set point. And that's very different from allostasis, where set points don't exist and they're irrelevant actually to the physiological functioning of a system. So I appreciate the call for specificity, but I just want to say that there's a huge literature out there, like huge, as in thousands of papers at this point, not all of which are at this airy-fairy abstract level. And if, you know, I would have, if there had been more time, I would have made some pretty specific hypotheses. I could have introduced them here, but not in 25 minutes. There's so much, you know, cursive knowledge that you have to have. I think in this literature, there are two problems, right? One is that all of these, you know, in situational robotics and cybernetics and, you know, control theory, there are vocabulary issues that make it hard to see when it's hard. You have to try to integrate across those to see what's similar. You have to integrate across language, you know, like what a state is different in different literatures. But then in a single literature like predictive processing, the same word can mean many, many different things. So for example, for some people, corollary discharge and efferent copies are the same, mean the same thing. And in some views, they're actually really, really different. So I think that we probably see the same kinds of problems here that we see, you know, that there's a little bit of exuberance with language that maybe gets in the way.
Paul, about predictive processing, making some very specific points. There's this person from, I don't know if you know, this person from Boston University, I think now retired, but Grossberg, I think is the name. and I think if you had read some of his work, you would have recognized a lot of predictive processing in there.
Yeah.
Okay, yeah, I mean, so far as I can tell, and I haven't looked at all the papers, not one of them can explain how we walk and how we walk at different speeds and over obstacles and so forth. Not one. They don't even say anything that comes close. And that's kind of disappointing. I mean, for something that's supposed to be as mathematically sophisticated as what Friston claims, it strikes me as odd that they can't explain that.
Well, I mean, there certainly are models of walking, including.
Yes, there are, but not Friston style.
Well, no, but you know.
That's the point, though.
Well, yeah, no, I'm not going to go and defend Friston, but I'm just saying that one should not dismiss a theory because it doesn't explain something that's important. You know, again and again, as I think Lisa said, you know, his is not the only version of many of these ideas, and some versions are much more at a level that I think you probably find more satisfying in terms of explanation.
Well, can we shift? Oh, sorry, go ahead. Lisa.
No, I'm sorry. I was just going to suggest a book. There's a wonderful book by Peter Sterling and Simon Laughlin called The Principles of Neural Design, and it was published by MIT Press in 2015. And although they don't really specifically use the predictive coding jargon, you know, they're really, the whole book is about, and they never use the word allostasis, even though that's partly what Peter Sterling is famous for. The whole book is about predictive processing in a metabolically tractable framework. And I think they talk about flies, worms, and some mammal, which I can't remember, but probably a rat, but I can't remember.
Paul, can I ask you a question, bringing back to the main things of the symposium, of the workshop, which is, I think builds on what Barb was saying, I think, and Barb can correct me, which is the way you describe it, it comes across very strangely, because in the following sense. It comes across very progressive. You tell a story of progression that for all of us here fighting against these notions of progress, it feels very, and then this came along and improved this, and then this came along. It's very, and it almost seems that at least the way you describe it, maybe it's the structure of the slides or something like that.
If I had another 20 minutes, I'd talk about some of the regressions along the way.
Well, right, but the way you describe it, I think, and it comes across also in the way it's written, I think, is that it comes like as these gradual increases of sophistication, and in a way that I think it undermines I think your points are brilliant, but I think it undermines that the way you're describing it, and I think I think that's only
because I need to limit, especially in a short talk. But I mean, I think what I'm proposing, and because I find the human lineage interesting, I also want to get there quickly. But I mean, some of the things that we see are actually consequences of the opposite type. So I don't really want to say progression or improvement or anything like that, but rather just transitions through niches, which have a sequence. And some of that sequence is retreating. Some of that sequence is, the retreat into nocturnality, which really made many changes in the mammalian brain that are very important for us to understand that brain. That giving up on vision to a large extent for 120 million years had a big effect. No? Okay, well, maybe Barb can say more about nocturnality, but my understanding is a lot of the reliance on vision and the circuits that in our ancestors were doing visual motor control was regressed in mammals. Is that not true?
We can't hear you, Barb. We still can't hear you.
Still muted.
Sorry.
Yeah.
The difficulty here is, well, first off, mammals per se, or the stem of sort of the mammal reptile, they didn't particularly go back at that point. But anyway, that's not even the relevant point. So the thing is that even a nocturnal animal is using much more vision than a fish. It's got much more things to control. It's got, even at night, more distance to see. If you think about the cortex of a rat or a mouse, half of it is devoted to visual representations. So the idea you've got to understand how rich a sense kind of terrestrial vision is, in terms of even nocturnally, how much more information you can get for that in terms of direction and localization and egocentric stuff than you can from any other kind of sensation, except possibly echolocation, which they're not doing.
Right, but the earlier animals and the amniote lineage that before we split off from the sauropsids were probably more reliant on vision
they may
than 120 million years later.
Yeah, but that's not really that's underestimating the residual vision. I mean, a massive amount of vision that can still be used to guide behavior, even in a nocturnal species. And very few species, will only exit and do things when no photons are present. So this distinction of sort of like darkness and light and complete mapping onto how much visual information you use, just I don't think is really correct.
Yeah, but I mean, we lost trichromatic, we lost the ability for a time for trichromatic vision. I mean, I'm not saying completely loss of vision.
I am I used to work in primate physiology, and I still do, in fact. So trichromatic vision is not color vision. Dichromatic vision is, sure. And it's you probably should think of that extra small amount of my colleague David Field calls it 10% more color acuity. It's not color vision. So animals are almost all diurnal mammals have blue versus yellow receptors, which they can make chromatic decisions. So there's I think by concentrating on just the most recent primates, we've missed the big sweep of what kind of visual information is out there to use and is used. And in some ways, primate vision is quite odd with, being highly foveated, loss of a lot of acuity in the periphery, specifically loss of, not just any, but all chromatic distinctions in the periphery. So if you take a kind of more detailed look across what the information sources and capacities are, I think we've been, I guess they blinded a bit by our own vision. We need to really look, I think, at what kind of visual information is used by all these different animals, not assuming that it's nothing if an animal is quote nocturnal. And
yeah, you actually, if I may, I here I would plug another paper in the special issue by John Kaz and colleagues, where they discuss specifically the visual system of primates, how it formed. I think that's a lot of interest for many of us primate physiologists, because they explain things like the origins of dorsal and ventral. Maybe others would be interested.
Yeah.
I also wanted to apologize that I unfortunately have to go, because I had a prior commitment even before we scheduled this thing. So I'm, much as I'd love to stay.
I have to go as well. So I want to thank you.
I'm going to propose that we honor the original end time of the meeting. And thank everybody for attending. That was really amazing. It was a really great set of talks. I learned a lot. And
I just want to also apologize. Sorry, I just want to apologize to all the people on the chat who posted questions. They were really great questions. We just didn't anticipate how good they would be, how many. So.
All right.
Okay.
All right. Thank you all. Thank you all again.